the blood pressure medicine that we use. I mean, there's a lot of blood pressure medicine can use for the treatment of uh, corona. I mean, COVID-19 as well. This paper published in 2020 by Circulation Research. Okay. In addition, recently our VA participated in COVID-19 trial on drug use in curb inflammation, because I had mentioned that the inflammation is also the one of the factors for COVID-19. A drug called tocilizumab or TCZ, often used to treat the rheumatic arthritis. This medicine is basically used for rheumatic arthritis by the lowering the inflammation level in the focus is the focus for the new clinical trial on COVID-19 patient. Okay, so as you know, this pneumonia triggers a hyperimmune response that can be more detrimental to some patient than the original infection. That's another problem. So TCG, as I mentioned, this drug is a class 1 drug, as I mentioned, that reduce the strength of the body immune system. An overactive immune system is often as harmful as one of the stop working, with the potential to lead many type of autoimmune diseases, a condition in which one's immune system mistakenly attacked by the body. So this drug is trying, but in our VS system, don't know yet. Hopefully it will work. So, and we don't know. Eventually, maybe other drugs will come and see, will help us. So this is the two drugs are now on the trial. One is the high blood pressure reducing medicine, hypertension medicines, and other is the rheumatic arthritis med drugs. So next is the new approach is now is a clinical trial is the development of vaccines. So the vaccine is just they're trying to do it a copy of genetic materials from a virus and add it into the artificial nanoparticles. Now they're making they're taking a genetic materials from the virus that cannot produce any symptoms of disease, but it can target to that and nanoparticles, and makes it possible to create a vaccine based purely on the genetic sequence rather than the virus itself. Why they are using the genetic sequence? You know that because this small piece of genetic sequence cannot produce the disease. But it is unclear if such RNA vaccines are potent enough to provoke a sufficient response from the immune system. It is not clear, because immune system will work in two different ways. Either it will react with the RNA vaccines that will create problem in our body, or the RNA vaccine cannot trigger the immune system to destroy the virus. So let us see. Still, this is under investigation and trial. Hopefully, it will work. In addition to that, the chloroquine, and which is a very interesting molecule. It has a lot of political molecules also. Typically used to fight malaria, might inhibit the release of the viral RNA into the host cells. And the favipiravir, a drug that from Japan could keep virus from replicating their genome. A combination therapy also they are trying with different combinations like blood pressure medicines, with arthritis medicine, with antiviral medicine. They're thinking in different way, different combination to treat the disease. And also some HIV medicine also they are trying. Nobody knows whether it will work or not. So we have to wait for them, and it's very frustrating, I know, because a lot of economic situation. Now, there's another question always patients are asking: the COVID-19 and mosquito bites. But interestingly, it has no effect in to the virus that causes the COVID-19 transmitted by the mosquito. The question is that everybody asks this question. A study of the Kansas State University Biosecurity Research Institute, the first to confirm that this virus cannot transmit it to people by mosquito bites. The study has been published by Nature Scientific Report, July 2020, and credit goes to Kansas State University. They did this work. Next part, and supporting the who, the studies, the first provide the conclusive data supporting the theory that said by Higgs, Pence, professor of the biosecurity of university. So now, next part is the very, I'm almost very close to my top. That is a pregnancy and COVID-19. Why pregnancy is, people are, physicians, scientists are thinking that what is the COVID-19 effect on pregnancy, any effect. So before I go, I will say one slide, I will show you that. That is the thing. The problem is that the multiple studies have established that confirmation vertical transmission of the COVID-19 virus from the mother to the fetus. The route of the infection is through the womb into the utero well before the onset of the labor and the delivery of the baby. That's the big question now, that whether the baby will be infected by this virus or not. Still, we don't know, but it is they found that the virus can transmit from vertically from mother to the baby. So still the research is going on, and again, it's so frustrating. I'm here to give some talk, but most of the time I say I don't know, I don't know. This and it's very frustrating. But so why? I have to give some ideas why we are concerning the pregnancy is very easy an issue in COVID infection. The reason is that, as I mentioned, discussed earlier, that this virus, COVID-19, destroy the blood vessels, produces abnormal angiogenesis, blood clotting, and a lot of factors that destroy the endothelial cells, which is the building block of the blood vessels. So now, if you look at the very simple thing that in womb, we need to see it is the first trimester. If you look at the pregnant, the blood vessel start, in a critical component of the healthy placental function is the proper development of its vascular network. Very important is the vascular network. If you look at here, the first trimester, 0 to 12, you can see very little blood vessels start the formation. And then if you go to the second trimester, you can see week 13 and 28, you can see the blood vessel formation start, and huge blood vessel here, highly vascularized things. So that means it needs the blood. So in normal condition, so you need blood vessels. And third trimester, you can see the maturation is a lot of completely blood vessel formation. We take it from the Bible. Okay. So in a normal condition, you can say it's okay. In a normal condition, we are getting 3 to 5% chance of having a baby with birth defect. There are no published studies to COVID-19 in pregnancy and birth defect. But based on, if you look at, that's I can say that the because look at now a little bit discuss about this about how the blood vessels work. Hello, can you hear me? Hello.
Yes, we are hearing you, sir.
Okay,
Yes.
I have some problem here.
No,
So
you're perfectly audible and visible, sir.
okay,
You're
so
perfectly audible.
now the why the scientists, researchers, and the physicians are thinking about the COVID infection in the pregnant woman, because as I mentioned that and they are thinking, preeclampsia condition is a abnormal blood vessel formation in the baby. So it can accumulate it. If the mother infected during pregnancy, then why is the echo is forming?
Yes, there is an echo now. Pranav sir, could you please? Look
Yes.
into the matter.
Yeah, I am looking.
Deliberately ignorant. You have been
I
deliberately
request
ignorant.
others except the main speaker to keep yourselves muted.
Yeah.
We have our own liabilities like loan, salaries of employees, and other extra.
There is a background sound.
For last 3 months,
For last
I
3 months.
request all audience to keep your systems muted, except the speaker. All systems should be muted. Lots of background sounds. All audiences are requested to keep your systems muted. All devices should be muted except the speaker. All devices
Yeah.
should be muted. Thank you.
Thank you. Okay.
I think, sir, you can. I
And
think you can continue.
okay, so I can go back to my previous slide. How can I go back? Okay. Okay, so another another part is that because of the way we are interested about the pregnancy and the things that the a high fever in the first trimester can increase the chance of the sudden birth defect. And if you have the corona infection, you will get high fever. And that much indirectly, you know, there's a birth defect in birth defects as well as some abnormal development of the baby. So that's why we are very much concerned about the pregnant mothers. They should avoid this kind of infection. But again, you don't have any choice. You don't know who will get infection. If you get sick in the COVID-19 or any other illness and develop a high fever, please speak to your healthcare provider as soon as the possible and discuss best way to lower it, because you can reduce the temperature, body temperature or fever. Okay, so our next slide is the COVID-19 and cancer. As I as I discussed earlier, that the cancer is also is a disease caused by abnormal blood vessel formations. Because look at the pancreatic adenocarcinoma. If you look at the pancreatic adenocarcinoma, they don't have very hypoxic condition. What does it mean? Because very few blood vessels are there. And all the blood vessels are abnormal, mostly abnormal. That's why when you give the chemotherapy or treatment, this is pancreatic cancer, we cannot treat them. No treatment we have yet. Because two reasons, because of the lack of blood vessels or abnormal blood vessels, and they have a huge fibrotic barrier that's called pancreatic cancer. If you look at then the breast, on the other hand, or lungs cancer or liver cancer, you can see there are a lot of blood vessels, highly angiogenic. But interestingly, none of the blood vessels are normal. They are abnormal blood vessels, like we found in COVID-19 transfected patients. So possibly they are same mechanism because we found a similar kind of gene patterns. A lot of almost 80% genes are same in cancer and COVID-19 transfected patient samples. So in this situation, it will may if you have after having the cancer, if transfected with the coronavirus, and if they survive, that's okay. But the chance of more abnormal blood vessel surrounding the cancer or hypoxic conditions surrounding the cancer, which prevents or destroy the treatment process. So you can say, in other way, COVID-19 infection may cause abnormalities in blood vessel and also prevent the treatment of the cancer or destroy the cancer treatment procedure. In addition, so people who have cancer getting cancer treatment often have a risk COVID-19 because their immune system is very poor, so their chance of getting COVID infection. So this will be very critical. We have seen a lot of uh, cancer patients. They died because of the COVID-19, infections. And another thing is that in COVID-19 and cancer, the weak immune system that control their body's different systems. Weak immune system because of that. And also, COVID-19 promotes the weak immune system. So that's why, the cancer patients are very prone to that. Then the damaged mucous membrane. What happened? The mucous membrane in our body, which form the moist pink lining layer of the mouth. If you look at the mouth, pink lining layer is mucous membrane. Throat, nose, eyelid, digestive system, vagina also act as a partial barriers against the infection. This mucous membrane is repaired and act as a barrier against the infection. And this membrane normally help protect us from the germs in the air we breathe, our environment, and in our food and drink. Okay, so cancer treatment, such as chemotherapy, targeted therapy, immunotherapy, radiation therapy, whatever the surgery, and certain procedures like putting in catheters or IVs or getting shots can injure cells in skin or cause damage to the skin or mucous membrane. All these factors that damage the skin or the mucous membrane, this makes it easier for germs enter in the body. So look at that. So damaged mucous membrane is another factor for the infection. Okay, some cancer itself increase the infections, like all the most of the blood cancer, leukemia, all the blood cancer or lymphoma, you know, they increase the infection in our body, the normal pathophysiological condition. So that's why they are very prone to this disease. Okay, so next, infection can increase the abnormal, as I mentioned several times, because I'm a angiogenic person, so I prefer always talk to a angiogenesis. So, but it is true that infection can increase the abnormal vascularization in the body. And after transfixion of the COVID, it also increases more abnormal vascularization. So failure of the treatments is very high. Okay, in addition to that, that some treatment can increase the infection, like chemotherapy. Like if you give some diabetic patients, diabetic treatment or diabetic patient, they are very prone to infection. So it also increases the disease. This makes it okay. And then finally, we have seen this one. This is already stem cell transplant. Sometimes they need bone marrow transplant or stem cell transplant. That also help COVID to transfect easily in our body. So altogether, so if you look at that, how this system in our body, the like pregnancy or cancer together, either or either they help the COVID to transfect in our body, or COVID transfection eventually destroy the treatment systems in our body. So we, because of the poor and increases the poor angiogenesis blood vessels. That much we know from the different studies, and we don't have any more data for currently about the COVID on their treatments or other things. It is very frustrating, I know, because you are thinking that we might get some good news very soon, but I don't know. Maybe end of this year or next year we might get. Really, we are expecting some vaccines, but don't expect too much from that. antibody therapies. Second phase of the antibody is crucial, which is very now. Anyway, so I want to finish my talk here, okay, because it is almost 2 o'clock now here. I'm very, very.
Sir, I'm interrupting again. Kindly keep to your right. Yes, sir. Thank you.
So now, so I have to acknowledge these people they who helped me to do all these things. Snigdha Banerjee, Namul Haq, Amlan Das, Suman Party, and other members of my lab who helps a lot to understand this disease. And we are trying to do some work on in our our plan to find out because the problem is that the COVID-19 in vitro studies people are doing here, but it is not very easy to do it. So we're trying to make some model systems to find out whether we can see the normalization can help from the COVID infections and all. So thank you for giving me this opportunity again. So, and if you have any question, please feel free to ask me. Thank you.
Thank you, Dr. Mohua. Please. Dr. Mohua?
Is not there?
I
Dr.
am here.
Mohua?
Yes,
Yeah.
I have unmuted myself. I have unmuted myself.
Yeah, please.
Yes,
Please.
thank you so much, sir. Thank you so much for a brilliant address and for informing us about a very, very pertinent issue. I mean, the entire world is traumatized by this global pandemic, and we all look forward to getting a solution as soon as possible. And you have made us aware of the increased risks of COVID-19 infections in case of and mortality, fatality in case of cancer, in case of pregnancy, and in any case where there is heightened angiogenesis. There's one technical hitch here that most of our participants are following your program in the YouTube live streaming, and there's a problem with YouTube actually, and I don't have too many questions. I can just ask you one question. Sapna Tiwari, she asks that how chemotherapy is involved in infection during COVID-19 in cancer patients. That is what she is asking here. That is the only question I've got. Others are saying that they have a lot of questions. They can't really reach us through YouTube live streaming. So it's completely a technical hitch. I'm very sorry for this.
Okay, that's fine. Yeah.
Yes.
No, that's okay.
So one question is about how the chemotherapy involved in COVID-19 infected cancer patient.
Okay, so couple of things. When you give the chemotherapy to the patient.
Sir, your microphone, please.
Okay, sorry.
Yeah.
So, are you okay now?
Yes, sir. Yes, absolutely.
Okay. The problem is the chemotherapy. If you give the chemotherapy, first thing is that it destroy your immune system. Sometimes, okay, not always.
Yes.
Sometimes it destroy your immune system,
Yes.
and that's why COVID-19 get opportunity to infect patient very quickly. And second thing is that the two way. If you have a COVID-19 infection, it disturb the chemotherapy treatment because, as I mentioned, that because it blocks the normal blood vessel formations, or is causing abnormal blood vessels, in the surrounding the tumor. So that's why the drugs will not properly deliver. That's another one reason. On the other hand, the chemotherapy itself creating a lot of immunological problems. So, like particularly, if you have a blood-related cancer, then the COVID-19 has advantage. Oh, okay, this is the good place to go and hijack the system, and all these things. That's the both way is bad. Otherwise, that's I know. I mean, so far we know that this is the way the chemotherapy and COVID-19 relation. But eventually, of course, if you destroy the mechanisms, eventually, then we can put it. Main thing is that we don't know the really, honestly speaking, that is, we don't know the real mechanism or whatever the mechanism. The only mechanism we know, the COVID can, the how to enter into the lung cells. But
Yes.
we cannot target this. This is not a druggable molecule. That's the problem. If we target
Yes.
these molecules, we will inflammation goes up, blood pressure will go high. So it may be you can destroy the virus, but then the patient will die because of the high blood pressures
Exactly,
and
because of comorbidities. Yes, absolutely.
comorbidities.
Yes.
Yes.
Yes. Yes. Yes.
Okay. So
Thank you very
and
much, sir.
again, thank you
I
for inviting me. And I mean, I don't know how much I can give you the new information, but I try to my best. And
It
still,
was very informative, sir. It was very informative, and we are very glad to hear from you. I'm very sorry about the technical hitches,
no,
but
that's okay.
we will actually upload this entire talk in YouTube, and anyone can actually see you. The full video will be uploaded later this evening by the
That's,
organizers.
that's okay.
Yes,
So,
yes, Prashant, thank you very much. I
so it
think
is
it is
already
morning.
1:27. I have to leave
Yeah,
because I have to go to bed. Okay.
yeah,
So
you have the work tomorrow. So thank you very
yes,
much.
yes.
Thank you very much.
So
Actually, I see so many participants in our Cisco platform also.
There are lots of
So
participants. There are lots of
lots of person.
participants, but yes,
Some technical problems in YouTube, but it is still showing connected. I think it is a transmission is going on. Anyway, Shrahamda, thank you very much. I have no language to thank you and giving so much of trouble to you.
no trouble. And I should because a lot of problem here. That's why I could not get enough time
Yeah,
to
yeah, yeah.
really make it more slides for you or more information. And maybe next time I can, if I get opportunity, I will.
Physically. I
Maybe.
want physically.
yes, sir. We want your physical presence in our university, in our department.
Okay, if we get
Sir,
favorable
there's one
advantage.
question here. Can I just ask
Sure,
one question?
sure.
This is a student, a research scholar from our department. Is Bishwajit Das. Bishwajit Das asks for the rapid test kit. The presence of IgG and IgM, immunoglobulin G and M,
Yes,
are
yeah.
detected in the subject's blood. Serum level of immunoglobulin G and M can increase in other types of viral infections also. So if a
Yeah,
person is suffering from normal influenza or is having fever or any other kind of infection, it may rapid testing kit as well, and the subject may be taken to the COVID care center where he might ultimately get infected with the COVID-19 from other patients. So his question is, instead of soluble antibodies, the presence of any other antigen directly related to COVID-19, could that be used for the rapid testing?
yeah, but you want to test for other disease or it interfere with the disease? Now the question is not you don't need to because it's expensive, right? For
Yes,
flu,
yes,
for influenza, you don't need this kind of kit because flu is now under control. Everybody knows that. Okay, so
right.
but for this, yeah, some you can, in other way around, you can think like the other way. So maybe the some data is not what you are getting. Maybe rapid test is not very authentic. But it is sensitive, but sometimes you can get false positive some data. Okay,
Sure,
that's a different issue because but you don't need to use for other disease like flu. But flu is
right.
next after 2 years, 2022. I'm very sure it will be like flu.
Yes,
We will live with the COVID whole life. Next, I mean, maybe you
like
cannot
that
get
is
the
HIV. Like the life is
like
still going
HIV,
on with HIVs.
like
Yes,
flu, it
like
will
flu,
same
like
that.
influenza.
Yeah,
Yeah. Yes, absolutely.
yeah. But you will get some medication, some medicine will work. But eat the healthy food, I mean, you have to stop something, you have to eat good food, good healthy food, and don't take cold water. Cold drinks are not good for now.
Yes, yes.
Yeah, I told my daughters, but they don't want to listen it, Oh, no, cold water is fine. Yeah. Okay.
Okay, sir. Thank you so much.
Thank you, Sharada.
Okay.
With permission from the host, may I start the next invited lecture?
Yeah. Please invite our speakers.
Yeah, yeah. So our next speaker is Professor Ariel Kushmaro. He is a full professor in the Department of Biotechnology. Yes.
You have to make him the presenter.
He's a Department of Biotechnology Engineering in Ben Gurion University, Beersheba, Israel. And Professor Ariel Kushmaro's research activities focus on the development of various techniques for environmental sensing, bioremediation, and environmental microbiology. His research is aimed at understanding the structure and function of microbial communities and their dynamics with regard to the environment. These, under defined conditions, these investigations include environmental monitoring of viruses and bacteria, the isolation of bacteria, and their study under defined conditions in the laboratory. Research projects include wastewater monitoring, treatment and uncultured bioremediation, microbial encapsulation technology, discovery of novel antibiotics from cultured and uncultured microorganisms. The group of Professor Kushmaro includes 10 PhD students, 4 MSc students, 4 postdoctoral students and technicians. So far, he has published more than 110 referred journal articles and has got several patents. And with this brief introduction, I hand over the platform to Professor Kushmaro. We look forward to hearing from you, sir. Please.
Thank you very much for the introduction. I hope you hear me and you see
Yes,
the presentation.
Loud and clear. Absolutely.
Yes,
Okay.
absolutely.
Okay, so thank you for the invitation. And it's unfortunate that I can't visit you in your lovely Assam state in India, but maybe in the future we can do that.
Yes, we look forward to that, sir.
And so we're staying globally and joining our thoughts and science together. So I will thank also to the previous speaker that done a very nice introduction. So I will continue and talk about how we can do early warning and pandemic tracking of COVID-19 using sewage and wastewater viriomics. Before that, I will make a very short introduction related to Israel and Ben Gurion University. So we are locating in the Middle East, and you see that Israel is a very small country. And here you see the map of Israel, and Beersheba is located in the south of Israel. The population of Israel is about 8.5 million, and most of the population are concentrated in the center of Israel, where is Tel Aviv, the area of Jerusalem, and less populated in the south of Israel and the north of Israel. Later on, I also mentioned the city of Ashkelon around here, that we done some kind of pilot study, but you will see a lot of names later on during the lectures of different cities that we done some wastewater analysis. So you will see Haifa, you will see Tel Aviv, you will see Ashkelon, you will see Jerusalem, etc. Okay, and another thing, I'm a researcher in the Department of Biotechnology Engineering at Ben Gurion University, and also heading the environmental biotechnology lab. That one of our projects is related to the corona. We're doing a lot of other activities. It's including discovery of bioactive molecules, bioremediation, marine microbiology, biofilm studies, and more and more. And actually, our interest in corona and SARS-CoV-2 virus actually started only in February, March, when the virus arrived to Israel and to Europe. And then we were starting to do some research concentrated on wastewater analysis. And the reason for that, because we've done it before, but on different pathogens, mainly bacterial pathogens, but also had several projects, including in Singapore, that deal with virionics of water. Do you hear me correctly?
Yes,
sir,
you are
proceed.
audible very nicely.
Okay,
Proceed.
because I hear some background noises, so maybe it's from my microphones. But anyway,
Yeah. Other
let's.
microphones are to be switched off or muted. Only the speaker is to speak. Only the speaker's microphone will be
It's
switched
okay.
on.
We will survive. We will survive.
Okay, thank you so much, sir.
It's okay, no problem. So anyway, when we're looking at the structure of SARS-CoV-2, that is the causative agent of COVID-19, we can see that inside the virus or the core of the virus, we have an RNA nucleic acid, and this is what's interesting us for the detection. And if we look, then we can see that we have a very interesting structure with the spikes proteins that also related to the name coronavirus. And again, when we're doing wastewater detection and we do it basically on the nucleic acid, we are more interested in the RNA that dictating the different genes that important for the virus. Now we have to remember that in the wastewater we have a mixture of this RNA because the source of this RNA could be the virus itself, but also it could be leftovers or decaying virus with cells and other components that could be in the sewage. If we're going now and to see the situation in the world, and this is updated to 27 of this month, we can see that the red zones are the places that the disease is very active and the spread is very active, and unfortunately India in a dark red and other region around the world. Israel is also in red, but I will say less strong red. And the situation is not good in terms of spreading of the disease, but in certain areas, even though the virus is spreading tremendously, the level of mortality is lower. I mean, certain countries in the Middle East and even I think in India, the mortality rate is lower than what we've see before in Italy, Spain, and now in United States and Brazil. So if we're talking about the mortality rate, it was estimated around 2%, but this is very tricky because we don't know exactly how many people are infected, who is symptomatic or asymptomatic, etc., etc. Also, the tests are limited, and usually in most of the places it's only for symptomatics, and people that they are asymptomatics, most of them will not test it and continue to spread the disease. And in certain places, they show like Italy and Spain that the mortality rate is around 10%. And previous knowledge was from the previous outbreak of SARS was 10% for SARS-CoV-1, and we know for the MERS it's about 34%. But then the transmission rate is considered to be lower. Now, another important issue that we saw also in the outbreak of SARS-CoV-1 in 2003, and in the this outbreak with SARS-CoV-2, that there is a shading of virus from the patients, symptomatics and asymptomatics, and actually that we can detect it in the sewage itself. Another interesting story that I want to share with you is what's happening in certain countries, and I choose to show you Spain and Israel, because over there I can pinpoint why there is an outbreak or why the outbreak started. So if you see in Spain, this is the daily new cases. We see that the outbreak starting about March 10, it's spiking very high, and then there is a decrease in the transmission, then it's very low, and now we see the beginning of the second wave of outbreak that might be very high. We are not sure yet, but the reason for this wave, it's the opening of the lockdown. But what's happening in this point, in this end of the week, was a lot of demonstration all over Spain, especially in Madrid, for the right of women. And it's a big festival with tens of thousands of participants in the cities, and especially in Madrid. And this is how it looks like. So a big crowd of people shouting, celebrating, and of course, this thing generated a big transmission event, and then the outbreak. Another issue, in the same weekend, it was very important football matches with 30,000 people in the stadiums. So this is also some contributing fact. So this is how looks this kind of demonstrations in the streets of Madrid. And obviously this kind of crowdness, it's very problematic, and it's emphasized the need for social distance. Now, the second example is coming from Israel. So we see below. So in Israel, the outbreak start around also 10, 11 of March, go up to a certain level, and then went down because of the lockdown. This is the period after the lockdown, and now we can see that around May, there is beginning of the second wave that now is skyrocketing, and it's quite high. Now it seems to stabilize, but now in Israel we're trying to survive without lockdown, and maybe some lockdowns in certain neighborhoods or maybe cities, but not all over the country. And now here, the reason for this beginning outbreak was a celebration in Israel that we call it Purim, and people going out to the street with costumes. There is a lot of religious ceremonies, and again, crowdness of people. And this was one of the reasons for the outbreak in Israel. So after I says all of this, Let's go now to wastewater story. So as I told you before, the SARS-CoV-2 is shade from people into the sewage, from the toilet, washing hands, and other kind of activities that you're doing in the washroom, and everything is going via the sewage to the wastewater treatment facilities. Now, if we can detect the virus in the sewage and relate it later on to population, it will be a very important step, because first of all, if you think about it, it's a pooling of a big portion of the population. In one sample, it could be a pool of 1,000, 10,000, millions of people in one sample. Another issue, if there is certain communities that don't want to test themselves, direct sampling from the health authorities, this is also could be overcome via the sewage in certain neighborhoods or certain areas. Another important issue, it's ethical, that again, it's pooling of virus for a lot of people, and it's not personal. So this is also an issue that we can do in consideration. So I will say that the best situation, it will be to take the sample close as possible to the source, because the degradation of the virus starting already in the human body and then along the sewage. So let's say it stay in the sewage system for several hours or maybe several days. This is also will influence the concentration of the virus, the degradation of the RNA or the virus particles. Temperature will be important, and a lot of other parameters like organic material concentration, if there is a source of industrial wastewater, if there is decontamination in the water like chlorine, other materials, all of those issues can influence in the end on the test. So actually, in a nutshell, we have the population, there is a fecal shading passing via the sewage into the wastewater treatment facility, and we can catch or sample before it's entered to the wastewater treatment facility, or maybe in the process, and then we can do the wastewater surveillance for the virus, and then according to the data that we'll supply for the health authorities, then they can do some outbreak response, and then I can, and soon I will show you some examples. So we published in beginning of May an article that soon will appear in Frontier after peer review process, and the title is Regressing SARS-CoV-2 Sewage Measurement onto COVID-19 Burden in the Population. And actually in this manuscript, we actually presenting a proof of concept for quantitative environmental surveillance. And you see that there is a lot of co-authors, some of them from the Ministry of Health, people from my lab, and other groups that help in the effort. And here we can see a picture of one of my postdoc, Karin Yaniv, that she's handling the samples in a BioLevel 2+ facility that we can manage the samples in the proper safety ways according to the regulation. Also, we're using different primers in quantitative PCR in order to evaluate the concentration, or let's say the copy number. So it's very popular to use the E gene or the N gene. E gene is a code for early genes of the virus, and N gene is nucleocapsid genes. Okay, so all of them are on the RNA of the virus, and the problem is that in sewage we get pieces of the RNA, and the virus is already passing some degradation process, and this reason you will see that in our detection method we're trying to use several genes, not just one. We usually will use the CDC target primers and probes, that is N1, N2, and N3. I will show you that we're using also additional ones that we develop in my lab, and also we're using some of the E gene that I just made a mistake. It's related to the envelope protein and not to the early genes. So anyway, this is the what's approved by the CDC and the WHO, and seems to be work, but you will see later that it's not work in sufficient way for wastewater detection. So anyway, we're using the technique with qPCR, and just very briefly, we're using primers, and together with Tachman probes that will later on will give us a fluorescent signals and improve the sensitivity and the specificity of the detection. I will not go into that and the technique of the RT-PCR. But what you have to understand that actually in the process we need to concentrate the sewage, we need to extract the RNA, then to do a process of reverse transcriptase that could be part of the qRT-PCR, and then run it with the primers, with the Tachman probe, and later on read the results, and this is what we get. So here we can see the threshold and the noise and everything that's going up here. This is the positives of what we can detect in the sewage. So this is how look the qPCR screen. Okay, so let's go for a little bit for the data. And I want to say that when CT, it's the threshold cycle, and I will go back one slide. So as the threshold cycle is smaller, it means that the concentration of the virus in the sample is higher. As the CT is higher, it means the concentration of the virus in the sample is lower. And actually, we're using in most of our results only CT, because it's very hard to go back and say the exact concentration, even though we have calibration curve and other things that we can say the amount of viruses per sample, but still might be some problem of inhibitors. And I will discuss it in a few minutes. So anyway, if we're taking now and checking on the raw sewage, and we prefer to use the raw sewage fresh as possible, and also it's simplify the test. The problem for that is that we are limited for early warning. So if the concentration of the virus in the sewage will be very, very low, and we're using only the concentrate, the raw sewage, it will not be enough and will not detect anything. So for that issue, we're working now on different filtration technique that we can concentrate 5 to 10 liters of sewage and improve our detection limit. So anyway, here you see a sample name that it's actually it's names of cities like Bnei Brak, Shafdan, it's the wastewater treatment facility in Tel Aviv area. Bet Shemesh, it's a city nearby Jerusalem. This is area of Tel Aviv that we sampled directly from the sewage, and this is another area. So what we can see, and this is in April, and you see the dates here, we see that in Bnei Brak, in mid-April, we can detect the virus, and this city also connected to the Shafdan treatment facility, and also in the Shafdan, we can see that there is a virus. Here we can see that the concentration is higher than what we see in Bnei Brak. Then it means that there is more sources coming to this treatment facility that they have the virus in them. Other area in Tel Aviv, for example, that also connected to the Shafdan, and also this area, it's also connected to the same treatment facility, we see negative. So it means that those areas are not contributing to the sewage. What else we done? We worked on different concentration methods. We try with PEG and alum and other techniques. And I can say that most of the techniques didn't give us the proper concentration method as we expected. We in the beginning was thought that alum will be the best one, but we saw that alum and PEG are almost the same, and it doesn't give us a very good concentration technique. And now we move more towards filtration. I'm not presenting this data, but I can tell you that dead-end filtration using certain dialysis membrane gives us the opportunity to concentrate the sewage at least 20 times with very good results. Also, and this is an experiment that we use with the alum, and we saw that we can improve the concentration, but still not tremendously. So now we are bending this kind of concentration methods, and again moving to more filtration methods. So here we can see some kind of proof of concept of detection, and what you see in this table. So you have the name of the place. If it's a wastewater treatment facility or other source, and the date of sampling, and if it's negative or positive, I mean, no data, it's mean that it's negative, and the CT. So what's very interesting here, that you see that in certain places in Israel, that if we're checking, this is the beginning of the outbreak. North of Israel, it looks clean. The area of Tel Aviv looks clean. The south of Israel look clean in the end of March. But as we're going to the end of March, and this is Sheba Hospital that concentrated the people that infected. So we see in the sewage of the hospital, we see it's positive. And also this is the Sorek wastewater treatment facility that connected to Jerusalem. We see it's also positive. And then as we're going along into the month of April, we see that most of the places, except several, are positive. In several places, like the city of Bnei Brak, we can also correlate some of the city to the number of positive people, and this is data coming from the health ministry. Also, what we done as routine, we're checking for adenoviruses and also MS2 phage, it's E. coli phage that's supposed to be in the wastewater, and this is give us some other, I will say, some kind of positive control that our data is correct. So this is, I think, very interesting figure. So here we can see the SARS-CoV-2 CT in raw sewage according to the quantitative PCR versus the number of positive diagnosed COVID-19 in Bnei Brak city. That it's located very near to Tel Aviv in the center of Israel. And actually we see that, that when we have low number of patients, let's say a little bit more than 1,000, the size of the city is about 2,000 people. So here we can detect about 1,000 people out of 2,000 people. And as the concentration with the dates into the month of April is going up, the amount of patients going up, and also the CT is going down. And if you remember, if CT is low, then it means the concentration, the virus is higher. So, and I can tell you that this CT is about, I will say, 100,000 viruses per ml, and here it's couple hundreds per ml, according to our curves, calibrated curve. So let's go now to another story, and this is pilot that we done in the city of Ashkelon. It's also in the south of Israel, about 45 minutes from Tel Aviv to the south. It's situated according to the sea. It's a beautiful city. You see several pictures. This is the Mediterranean Sea. So there is also high building and low building. And actually what we done together with a company, Can-Do company, that they are specialized in sampling wastewater, we choose a certain junction in the sewage system all over the city that actually concentrated several neighborhoods. So we see this is one neighborhood, second neighborhood, another neighborhood, another neighborhood, and each one we have a sampler that can sampling automatically for 24 hours. And actually the idea is we're taking the wastewater sample, it's combined, then we do the analysis as I show you, and then we look for indication for infected people in the population, and this is some of the people that also participate in the project. So the first one issue was the sampling. So here you can see the automatic sampler, and together with the automatic sampler that's sitting in the manhole of the sewage, there is also different sensors, including flow sensors, turbidity sensors, COD sensors, etc. And here is samples coming out from the samplers, and this is shifted to my lab, and over then we done some analysis. So I'm not going now to show you numbers, but what I want to show you which date we took the samples. So this is the outbreak in Israel that started in March, and around this area the lockdown started, and then was decreased, and end of April, beginning of May, they released the lockdown, and life starting to go back to normal. And this is now the infection rate in the city of Ashkelon. So we see a plateau here. We sample around this area, 17 of May and 26 of May. And a little bit later in June, July, the outbreak starting again because of release from the lockdown. And actually, you will see that during sampling this period, actually we predicting the increase of the virus in the sewage, and actually we see in advance of several weeks this kind of outbreak. So let's go to the preliminary results from the 17th of May and 25th of May. And this is the different neighborhoods and sampling location in the city. And actually, here you see the relative amount of virus in the sewage, or what's say in the neighborhood. So yellow is very, very low or none, and red is high number, okay? So when we check the 17th of May, we see that there is several neighborhood in yellow and some in orange. So it means low infectivity. The yellow, it means no infectivity. And here we can see a red area. So this is what we call in Israel Corona Hotel. It's a place that they put people that they have no symptoms or maybe mild symptoms in this kind of hotel, and they have they're isolating them there. So they are also source of virus into the sewage. And what's interesting that after 1 week, we see that all the city is already infected, and certain neighborhoods, especially this neighborhood, is a strong red. And also this neighborhood seems to be with high numbers. Okay, so according to this, we can take a city or an area and say what will be the risk of each area, and decide where to do a lockdown. For example, if the authority will ask me, I will tell them in the end of May, here you have an area that you need to do a lockdown, and maybe other neighborhoods you can do it partially. So this, I think, very crucial information, and now we planning to do it with the company of Kandu and the Ministry of Health. We're trying to do it all over Israel, including hospitals, certain neighborhoods, and maybe also go globally with this kind of technology. So another small thing before I'm finished, we are not satisfied with the primers that suggested by the World Health Organization and the CDC. And actually we took, and here we see this is regular PCR, this is the CDC primer, this is improved primers based on the CDC development, and this is new primers that we developed. And actually, you can see, according to the density, that the new primers and the improved primers are working a lot nicer. And I will show you this also in qPCR, and here we see examples. So this is N3 that is according to the recommendation of the CDC, and this is the log copy number of the virus RNA. So here we see that up to log 2, we can amplify with the CDC, but if we're using with the improved primer and the new primer, actually we can go 2 logs higher in the sensitivity. So I think this will be very important, especially in wastewater, if we want to use it as a early response and early alert for infection in the community. And this will be very important in area that are clean, and we want to see if infection is starting in this community. and this way we need a very good sensitivity. I can tell you also that in certain cities that now we analyze them, including the Tel Aviv metropolitan and other cities, we see that during the lockdown, there is decrease in the virus in the sewage till it's going to a low level. And when they stop the lockdown, then the concentration of the virus is going up. So this is why we need the sensitivity of the method. So here I will try to show you a movie that summarizes most of the things that discussed before I will finish.
An early alarm bell for sudden spikes in the virus. Currently, Israel is struggling to increase the number of daily coronavirus screenings, but even when testing upwards of 12,000 people per day, asymptomatic carriers are going unseen. And we now know that anywhere from 25 to 70% of virus carriers are asymptomatic. So, according to the Health Ministry's Dr. Itai Bar-Or and media reports, we can simply test the amounts of virus shed in wastewater and predict how many people are infected regardless of symptoms. And apparently, with initial tests showing very promising results, over a dozen teams around the world are trying out this same thing.
Management, so I would thank my group. I thank especially to Karin Yaniv that she's a postdoc leading the project. And also I would like to thank Satish Lakakula, that is a postdoc in my lab, that also joined the project in the last month and helping a lot in analyzing sewage samples from all over the country. So thank you very much, and I'm opening for question if relevant.
Thank you so much. Thank you so much, Professor Kushmaro, for a scintillating talk. There are lots of questions. Like Paramita Bhattacharjee, she asks, what are the chances of transmitting COVID-19 through sewage as they have already undergone various processes? So that is her question.
Okay. So first of all, it's a very good and knowledgeable question. And the answer for that we don't know yet, but most of the indication are pointing out that wastewater are not infectious for COVID-19. Probably what we are detecting is the leftovers and remains of the virus. There is no single study till now that show infectivity from wastewater. There is several studies that can show a possibility for infection from stool, I mean fresh stool, but not wastewater. And we have to understand that wastewater are also diluted, and there is also degradation that starts in the human body, and then degradation in the sewage itself. There is a lot of decontamination agent in the wastewater, including chlorine, soap, and other detergent that effective this kind of outcome.
Okay, thank you, sir. There's another question. This is Shunanda Basu from Ranaghat College, West Bengal. She asked that can we use the toilets in our college? If yes, what precautions should we take after reopening of the college after the lockdown is called off?
Okay, so if you ask me, just keep it clean. I mean, use it, clean it frequently with chlorine.
Yes.
And we know it's helping. And again, we don't know, or there is no indication for infections from toilets. I mean, if you keep your hygiene, washing hands and everything, then probably we will be okay.
Okay. Deepu Barmun, he asks, why do other animals have lower chances of being infected with COVID-19 compared to humans?
I think it's a very interesting question that I have no idea how to answer it.
I guess it has something to do with the receptor.
I mean,
yes, the
it might be,
angiotensin
yeah,
converting
I know,
enzyme
but
receptor 2.
I think it will be very interesting research to do, and I don't think we know enough about it yet.
Exactly. Yeah. Okay. Lots of comments.
I have one query.
Sure,
So, madam.
please.
Actually, some laboratories are using only amplifying the E gene for a detection of coronavirus 19. How much it is perfect to determine the COVID-19 by just amplifying the E gene?
Okay, so E gene could be okay for clinical samples that from the throat, because there is very high concentration of the virus there. But from our experience, using the E gene is not sensitive enough. N gene is better, and even N gene for wastewater, we don't think it's good enough.
Okay, for human samples, only E gene is not perfect, you are telling?
It's not. I will not use only E gene. I mean, I will use combination of E gene and N gene if you want to be on the safe side. Because with the E gene, according to my knowledge, there is a lot of false negative results.
Can we check also the non-structural protein genes?
It's possibility, but there is no, till today, there is not good primers for that and probes, as I know.
Okay, yeah. No primer, no universal primer is still detected for that.
Yes.
Anyway, Professor Kushmara, thank you very much.
There are lots of compliments from your audience and from the viewers. Your talk is highly appreciated by all the participants. Thank you. We look forward to having your physical presence in our department in Assam, and we look forward to more interactions with you and having you amongst us in the near future. Thank you very much.
Thank you very much, and good luck with your conference.
Thank you so much.
Thank you very much, Professor Kushmara, for giving us some of the valuable times.
Thank you very much. And unfortunately, I have to leave, so I will not participate.
We understand.
Yeah, we will be see you in other occasions.
So thank you for your time, and bye-bye.
Thank you. Thank you. See you.
Thank you, Professor Majumdar, for this opportunity you have conferred and entrusted me with. Thank you very much. I think we can go to the next session, which is, I guess, recess. And then we'll have our other technical sessions.
Thank you, speakers of all this session, and especially Dr. Mohua for comparing all the lectures and all the participants giving their patience time. So we'll take only 10 to 15 minutes time and we'll come back to the session again. Thank you. Thank you all. This one is
Professor Majumdar. Professor Majumdar. Professor Majumdar, can you hear me? Professor Majumdar.
Ma'am, I'll just connect him. Just a second, please. Dr. Sharnakar, I'll just connect or inform Professor Majumdar. Just a second. Kindly bear with us.
Hello, Dr. Sharnashika is there?
Yes.
Hello, madam. Okay, madam, we will take just 10 minutes break. Okay. And please, you also have a cup of tea from our side, and okay, we'll start. Yeah.
Okay, it is due.
Yeah, it is partial.
Personally, you will have.
Unless we meet physically in our university, then you have to bear with us and have cup of tea virtually from our side. Thank you very much. Okay, okay.
10 মিনিট ব্রেক দিয়েছি ওদের এক কাপ চা খেয়ে নাও আমাদের দিক থেকে
Dr. Yasmin? Dr. Yasmin? Dr. Yasmin?
Yes, sir. Yes,
Oh,
yes.
okay.
Yes, I'm here. I'm right here.
Okay, okay.
I
Okay,
just went to
so
have a glass of water, actually.
yeah. Okay. So just now it is it is 12:54. By
Yes.
12:05, sorry, it is by 1:05 we can start.
Yes, definitely. I'm here only.
Okay,
Start
okay,
with you.
okay.
Yes.
Whether it is this transmission is okay?
Yes, it is okay now, sir.
Okay.
Right
Actually,
now, there
dear,
is no problem.
doctors during doctors
Energies.
energy lectures, some problem are due to his laptop probably because some sounds are not clear sometimes.
Yes, yes. Yes, sir.
But Dr. Kushmar was totally clear.
Yes, sir. Sir, I will just
And
take this call by from Dr. Bhattacharji. She's calling me.
okay, okay, yeah.
Hello.
Yeah.
আরে আমাকে দেখতে পাবে, তাহলে দুজনে নিজে নে। এখন শুধু এইটা অন কর।
ওইটার কাজ হলো।
Yes, Dr. Yasmin.
Yes, sir. Shall we start, sir?
Yeah.
Okay.
Dr. Swarnakar is ready, I think.
Yes. So, good afternoon, everyone. I'm happy to welcome you to the second technical session of this webinar. And we are very pleased to have an eminent speaker as our first speaker for this session. I'm very happy to introduce Dr. Snehasikta Swarnakar, Senior Principal Scientist and HOD, Infectious Diseases and Immunology, IICB, Kolkata, India. Dr. Swarnakar secured a PhD in 1993 from Jadavpur University, following which she moved to the United States for her postdoctoral work, which she completed at the University of California, Davis, and the Marine Biological Lab during 1994 to 1997. She started her career in 1997 as a research scientist at the Stony Brook University, where she worked until 2001. And on her return to India, she joined the Division of Cancer Biology and Inflammatory Disorder of the Indian Institute of Chemical Biology, where she currently holds the position of a senior principal scientist. In between, she also visited the Scripps Research Institute in 2002 and the University of Connecticut in 2007. She is also a visiting professor at the Department of Environmental Science of the University of Calcutta. Dr. Swarnakar is an elected fellow of the National Academy of Science.
আওয়াজ আসছে না, তোকে দেখা যাবে না, তোকে এই অ্যাঙ্গেলে আছে, তোকে এই দেয়ালটা দেখা যাবে। আমি বলছি আমি এখান থেকে দেখতে পাচ্ছি, তুই শুনতে পাচ্ছিস তো? এখনো আসছে না।
Dr. Yasmin.
Some problem in connection seems. তোর কথা শুনতে পাইনি। তোর কথা শুনতে পাচ্ছে না নাকি? আবার অন করতে হবে নাকি? না না এখান থেকে কিছু আসছে না।
Dr. Yasmin. Dr. Yasmin. Am I audible, madam?
Yeah.
Am I audible?
Yes, now you are audible.
Yeah. Dr. Yasmin is not audible, you see.
Do I start now?
Yeah, Dr. Shondakar.
Hey.
Yes, madam. Madam?
Yeah.
Madam, am I audible? Yeah.
Now
So
I'm-
I think Dr. Yasmin is out of network now. You please start.
Sure. So I'll show my slide. Okay.
Yeah.
Okay. So before that, at the very outset, I really like to convey my sincere thanks to Professor Majumdar for inviting me to this web seminar, and also the whole team, those who are trying their best to connect all the people around the world. And the last two lectures were really very good, and we are learning, and at the same time we are adding some new information to the existing knowledge, which is really good for this situation. Let me start with my slides. So, as the title of my talk is okay, let me share. Okay. Hold on. Okay, I'm trying this one, right? Where is the text and things? This one. There is okay. Let me check how I can share my slides.
You go to the screen share option.
Okay, screen share.
You just here you see that in chart after before cross you will see the more options.
Okay.
In more options you go and share / sharing.
Screen. I will go to the screen. Share screen.
Yeah, in your screen you're seeing that symbol of cross symbol. Before cross symbol, 3 dots are there.
Okay.
In 3 dots, you
Okay.
go to there, you will get the more option.
Oh, okay.
Okay.
Yeah.
That upward arrow. Ma'am, that upward arrow, you have to click that. In a box, there's an upward
Okay.
arrow icon.
Yeah, share content is there. Also, from here you can there.
The third one, yes.
And then?
And then just click on that. That is the share content icon.
Okay. Just a minute.
Good afternoon, everybody. I'm sorry for the ma'am. To start
I'm
your
trying to share.
yes, ma'am. Yes, ma'am.
Madam, from left side, third key, share content. First one is the speaker symbol, second one is the this video symbol, third one is the share content.
Yes, the upward arrow in a box. Yes, ma'am.
Hi. Can you show the first question?
Yes. Yes, it's okay.
Where they are located? It's different. Yeah.
Can you see the slides?
Yes, yes.
Can you see the slide?
Yes, yes, yes.
Okay. So which slide is that one? Okay. So my heading today to talk is important strategies to manage and combat.
No, you have your slide share, madam. You again go to your slide sharing. Your slides are not coming.
Not okay.
Go to the slide share option.
The slides came and then they went away. So please click on that icon once again, the third icon. Yes, the upward arrow inside a box. Yes.
Left from the left, third one. Yeah.
Yes, from the left, the third one. Third one.
From the left to third one. Yeah.
Yes, ma'am. Yes. Thank you.
Now it's coming.
Yeah. Is it show?
I cannot.
It's gone again.
Coming or not?
No, ma'am, not yet. No, we can see you now. The third icon has to be clicked. The share content icon.
Yeah, share content. I'm going there. Then it is asking for screen. Click screen or application. So there
application. You go for the application probably.
Okay, application.
Do you lower bandwidth local computer condition?
And then
you go to the share content. Then you screen share.
Share content.
Ma'am, you have to remain in that share screen. You have to remain in that PowerPoint.
Okay. Remain in the PowerPoint.
Yes, what I mean to say is that yes, the share content icon. Click on that and stay there. Please. Uday, can you do anything from your side?
It will not come so fast.
Huh?
No, it doesn't come yet.
We cannot share content. Make sure that you are allowed permission to share content and then try again. Slowly. Light. Okay, what I can do, uh, I just redo the process and hopefully the screen can be shared. Huh? Just give me 2 minutes. No, no, আমি এখন just off করে দিচ্ছি। হ্যাঁ, কেন? হচ্ছে না কেন?
Pranabda, can you hear me? Professor Majumdar, can you hear me?
Yes.
Mohua here.
Yeah,
Can you
tell.
please ask the expert to send you the presentation. You can actually, as a host, you can present it and she can speak. I mean, the PowerPoint. If you show the PowerPoint,
In our
you
mail.
can just say next slide. Yes, next slide, and then you can just change the slide. Yeah, you please do the needful. Because yes.
You should send the PPT. We can.
Yes.
ওদের স্ক্রিন হয়ে গেছে। ওদের স্ক্রিনটা সরাতে হবে বলে নাকি? ওকে অ্যালাউ করতে দিতে হবে আমার স্ক্রিনটা। আমি তো স্ক্রিন শেয়ারই করে দিচ্ছি। হ্যাঁ, পুরো স্ক্রিনটাকে শেয়ার করতে বলছে তো। হ্যাঁ, শেয়ারের পাশে ক্যান্সেল আছে। ক্যান্সেল টিপলে তো ক্যান্সেলই হয়ে যাচ্ছে। এখানে লিখছে উই ক্যান নট ডিসপ্লে ইয়োর শেয়ার কন্টেন্ট, মেক শিওর ইউ হ্যাভ অ্যালাউ পারমিশন টু শেয়ার কন্টেন্ট। তো শেয়ারই তো করছি। শেয়ার করে কোথায়? শেয়ার করে অ্যাপ্লিকেশনই তো করছি। তখন কি চাইবে কোন স্লাইড? না না, ও নিজেই হয়ে যাবে, তো পুরো ডেস্কটপটা ওদের সামনে চলে আসবে। তাই? হ্যাঁ,
ম্যাম,
শেয়ারটা
আপনি
বুঝতে
আপনি যদি
পারছি না,
এটা
শেয়ারটা
হস্টকে
তো
পাঠিয়ে
সিলেক্ট করা
দেন,
যাচ্ছে
আপনি
না, শুধু ক্যান্সেলটাকে সিলেক্ট করা।
if I
হ্যাঁ
may interrupt, madam.
হ্যাঁ শিওর। হ্যাঁ হ্যাঁ
হ্যাঁ, আমি একটু বলছি যে যদি আপনি if you send the PowerPoint presentation to the hostকে পাঠিয়ে দিলে ওনারা একটা একটা করে স্লাইড দেখাবেন.
হ্যাঁ
You
হ্যাঁ
can
হ্যাঁ
give
হ্যাঁ,
your
দ্যাটস ফাইন,
lecture.
দ্যাটস ফাইন,
You can just say
ডু দ্যাট।
you can just say next slide and then they will change the slide for you. আমার মনে হয়
ওকে
সেটা সুবিধা হবে কারণ
ওকে ওকে,
নেটওয়ার্কের তো সমস্যা হচ্ছে.
হ্যাঁ ওকে ওকে শিওর।
ওকে, yeah.
আমার স্লাইডটা ওদের পাঠিয়ে দিতে হবে অ্যাটাচ করে। গুগলে আচ্ছা, এটা আরো ভালো। পাঠিয়ে দেবো, ওরা শেয়ার করে। হ্যাঁ হ্যাঁ, কোথায় যেন আছে, দাঁড়া এইখানে যা, এটাই যা, ওইটা ফরওয়ার্ড করে দিও ওদের। হুম। এই যে এটা।
মানে সব পাস দিলে না আর তোমার একটা দ্বারা আমি দেখি একটু সিনিয়র
Yasmin, Yasmin,
Yes, sir.
can we make the next speaker ready? You talk to Madam Swarnakar. If she takes some time, then we can make a next speaker.
Okay.
Madam Swarnakar is whether you can talk to her.
Good afternoon, Swarnika ma'am. Ma'am, are you able to hear me, ma'am? Swarnakar ma'am, are you online, ma'am? PBM sir, should I try contacting the next speaker and ask if she's ready?
Ma'am,
Should I
yes, ma'am. Next speaker.
should I contact the next speaker? Okay, yes,
Yes,
sure,
ma'am.
I will do that. Yes, yes. Give me 5 minutes. Let me try to contact her over phone. Thank you.
Okay.
Hello. Start my video.
Yes, yes, madam. It came. It came.
Very good. So you please show the slides and accordingly I will talk.
Okay. Enable editing. Okay. Let me share my screen.
And good. It came. Very good.
Very
Okay. Accordingly, you please change. Okay. So
Yes. Okay. Okay.
the topic today is important strategies to manage and combat COVID-19. So we have already listened to speaker, my morning speaker Sushantada, as well as the formal staff. So please go to the second slide. So second slide. Next slide, please. Next. Great. So when I please do that, okay. And as you can see, there are so many proteins on the envelope of the. Basically, the major one is the spike protein. It's a big one, and it's a transmembrane. And in that membrane, which is lipid bilayer, as you know, to do the protection, people say wash your hand, means you use your detergent so that it can solubilize the lipid membrane. In that lipid membrane, we do have spike protein, we have membrane protein, which is also inserted. The other two color that you can see, one is orange color, that's nothing but envelope protein, and the other one, the violet, which is hemagglutinin esterase. So those protein are on the outer membrane. But as we know, this is the positive strand, is a single-stranded RNA, and that's also having the capsid protein, which is nucleocapsid. So that's inside the core, which is the RNA that is going to be replicated when it's get the host. So next one, please. Next slide, please. Next slide, please. Can you go to the next one, please? Go to the next slide.
Yes, ma'am. Yes, ma'am. We are trying. Actually, this one is getting hanged.
Yeah, in the solar corona that we have seen, just like that outside the sun, the coronavirus also looks very much under electron microscope. Spikes, the protrusion outside the membrane, and very much like circular protein. So under the microgram, it's very, very small, and we need electron microscopy. So why we feel sick after virus infection? Because virus is not living until unless it enters the system, means some biological cell, then only it become living. So it relies on hosts to make copies of itself. And in response to virus infection, the human body activates the immune system. So once the immune system start activating, then the body start fighting against the virus. And when it starts fighting, then it needs a lot of resources from the body. So that's why with time it drains the resources and triggers inflammation process. And during that time, we do have fever. There are so many different viruses. Corona is one of the kind, and this coronavirus is RNA as their genetic material. There are viruses which is DNA. So those virus, those DNA and RNA acts as a blueprint. And this virus hijack our cells machinery to package its genetic material. So that's why while it is using our system, so our system get exhausted, and that's why we become sick. And the new virus then start propagating inside the cell. When the number inside the cell more than 100, and it burst out the cell, spread out, and then it infect the next cell. So please, the next slide. Why this COVID-19? COVID-19 is the pandemic disease, because earlier who thought of to give the name 2019 COVID? Then later on they think it's better to name COVID-19, the disease name, and the causative agent, as we have already heard from so many people, and we also came to know, is SARS-CoV-2, because already SARS-CoV-1 20 years back we already faced, and the similar kind of virus, which is MERS, that's also we have seen. So the outbreak combines twofold. One, it is a very emerging infectious disease, and this new virus, this SARS-CoV-2, is very contagious, means the disease is easily spread from cell to cell and from person to person. It's transmitted from one to other. And most people don't have the immunity because we never face it. So that's why system needs to recognize the antigen or the new invader. So that's why we never faced it, and this is one of the major cause for the outbreak. The next one, please. How does someone get rid of the virus? First of all, our body always tries to produce the immunological reaction when it gets exposed or get infected by the virus. But if a person has previously infected or vaccinated, their immune system will be primed to remember the reaction and will quickly make the right defenses, like produce the T cells. But if person has never been infected, then the virus has not been vaccinated, then the body's immune system uses all the tools to find the right defense. And that takes time. And during that time, when immune system get boosted up, during that time, virus start replicating. So that is also a balance between the virus and the immune system. And as I told, it's very, very small, and it's nanometer in diameter. And the spike protein is also pretty big, and it's not only one, the trimer, and the spike protein also has different domain. The outer domain where the S1 part of the spike protein. So people are now trying to catch hold on the particular sequences of the S1 domain of spike protein. Okay. And that spike protein is the one people are thinking of making antibody against it. So you can see, can you go to the next one? Next slide. Next. The next. Yes, please stay on it. So in the right panel, you can see the picture of a bat. So the COVID bacteria virus is also found in the bat as well as pangolin, and this has very similarity in the sequences, gene sequences, with the bat for 96%, and the pangolin cove also has 91% similarity. But the earlier SARS-CoV-1, which has similarity 80%. So that's why people are still thinking whatever the medicine they use during that time, SARS-CoV-1, similar, some of those medicines they are now using to tackle the new one, CoV-2, and sequence similarity is not up to 100%, but 80%. So there is still the question whether the medicine for SARS-CoV-1 is really working for COVID-2 or not. And if not, that's why we think for new vaccine. And other common coronavirus, which is in our environment, which is here for a long time, that has 50% genome sequence with the new COVID-2. This single-stranded RNA, there you can see in the right panel. So the different proteins that's there in the sequences, in the codon sequences, but this RNA is a long stretch, is 30 kilobase long, and there are 27 to 24 different proteins, and also open reading frames are there, 10 to 14 in the picture you can see. And in the open reading frame 1A, where we have polyproteins, and as in the virus outside the spike protein, that's the sequences are also there. So in each one particular virus, there are like 2,000 copies of membrane protein, 1,000 copies of nucleoprotein, and 20 copies of envelope protein. So all these different protein in different copies need to produce to make the perfect one single virus. So once it is getting entry inside our system, maybe that takes viral entry into cell is 10 minutes, and the eclipse period is 10 hours, and the burst size is 10 to the power 3, means 1,000 millions. So could you, and another thing I will tell, the spike protein enter into our mainly lung, and the receptor present in the lung epithelial cell, which is angiotensin converting enzyme, the binding affinity, the KD value for spike protein with the receptor EC2 is about 1 to 30 nanomolar concentration. Please go to the next one. So host cells, you can see our host cells is basically lung. In our there are two different kind of pneumocytes, type 1 and type 2. Both the cells has this receptor EC2, and it can interact with the virus. And alveolar macrophages, mucosal cell in nasal cavity, and those cells we do have, and the virus can infect. And those entry point, the nasopharynx and throat. So during detection, people also look at in their swab in the how much amount of RNAs are there. So anti-RNAs is 10 to the power 6 to 10 to the power 9 RNAs in the swab. Then only we know that they are positive, because nowadays we know how much RNA, and we do have a positive RNA, and then we are taking the sample from swab, and then we are doing the real-time PCR check the RNA concentration. So the RNA concentration, if it is to that level of 10 to the 6, and the reference positive control and the sample, and there we check the positivity if the amount of RNA is up to that limit. So please go to the next one. So what's their life cycle? Unless we don't know the life cycle, we cannot catch hold at which step we can block. Because today I'm going to talk how to manage this COVID-19 pandemic. The strategies to manage. So once we know the different stages of their life cycle, we can try each and every point for stopping the virus progression. Just a quick look. First, this virus is attached to the cell surface. So after attachment, and this attachment is through S protein. As I told, spike protein is one domain. And there is another thing we should know. In that spike protein also, there is one S2 domain. In between the hinge domain, there are few amino acids. So virus to virus, that sequences also change. And that's why it's a big challenge. The virus that we are now having, 2 or 3 different kinds, we make antibody, we make vaccine. That vaccine may not work next if that virus get mutated, because this virus has tendency to mutate. So in that S1 protein, which interact with the host cells, in the figure in the left side, you can see the virus is interacting with the receptor. There are 2 different receptors. One is angiotensin converting enzyme 2, and the another one transmembrane serine proteases, TMPRSs. So in that interaction time, people are also trying to stop by using some inhibitor. Okay, so in the entry point, once it's endocytosed, then it is fusing with the endosome, and then after that the free RNA is coming out. So sometimes the virus directly fuse with the membrane, and then RNA comes out into the cytoplasm. Now once the free RNA is there, then that RNA start using all the machinery of host and doing the production of different proteins, as well as the RNA also makes more RNA. So that's why the translation goes on. The proteins are produced. Some of the proteins get clipped through proteolysis. So during that time, proteolysis, there are host protein as well as virus protein, both are active. So people are also trying to use some protease inhibitor to stop the growth of those virus. So in the entry point, we can use some inhibitor. At the entry point, that receptor ACE2, we can make some soluble ACE2 in such a way that virus will not bind to host ACE2, rather it will bind to the soluble ACE2 that we are giving as a drug in the system. And when it starts doing its RNA to RNA, means replication by RNA dependent RNA polymerase in small form RT. So there are now drugs which is remdesivir, which is using these days also for COVID pandemic, and favipiravir. Those are basically inhibitor of RNA dependent RNA polymerase. Again, there are other points people can also think of, because once those all different component of the virus, like envelope protein, then capsid protein, and RNA, all are ready to assemble, means virus is another copy of virus. During that time, we can also use some inhibitor. Okay, and can I go to the next slide, please? So in the life cycle, we have seen the attachment, the entry, the replication, encapsulation. Means all those components are ready. Now they are assembled in such a way that it RNA material will be encapsulated, and on the membrane those protein will protrude out. And then the virus will have quite big number, like 1,000 of bacteria, a virus inside our host cell, one host cell. Then the host cell will burst out and those virus will come out. And that is the release. So these virus progeny are transported via Golgi vesicle, because that Golgi membrane, Golgi vesicle is also of the host, and virus will use all those host conjugation membranes, and then ultimately it will come out through exocytosis. So all these different points of the life cycle of the virus, people are now thinking to use drug for each and every point. And sometimes if the disease is very mild, then may not need the all different cocktail. And as already our speakers talked about, what are different symptoms. Initially, some people may not have fever, some people have fever and lung breathing problems, some people have no sense of the aroma, because that's one of the major symptoms. So depending on the symptom, people, the medical doctor prescribe different drugs. And as I told, all this life cycle in different points, those are the inhibitors people are using to combat the disease. So next, please. Clinical progression and transmission. So this is one of the major problem. That's why it's pandemic. So it's transmitted. From one person is going to another person, because it through seizing, it's coming to the air and then it's spread. I will show you the slide also. So in this clinical progress, what I'm trying to emphasize, that virus, when it is exposed, when a new person who is not having corona exposed to the virus, it takes like 5 days incubation period to have the symptom. And in some cases, when asymptomatic, for them is like more than 5 days. And the recovery in mild cases is 2 weeks. Okay, and for severe cases, it's 6 weeks. And the infected fatality rate so far is like 0.3% to 1.3%. So on an average, 2%, not more than that. And virus on this SARS-CoV-2 often transmitted respiratory droplets, and that's produced by coughing or sneezing. And this CoV-2 RNA has been detected in various surfaces also, even for several weeks. That's why the WHO or in the guideline, they always mention, and definitely we will also follow, wash, because we don't know which one we touched, when we touched, if anybody asymptomatic or symptomatic touched that area and virus stays for a while. So just to remove, we have to, because it stays for a while, wash intermittently our hands and face. And there are 3 different kinds we have already seen this virus. Still another new will come or not, whether it's mild or virulent, that's also a big question. So hand washing is kind of supportive way to prevent. Next, please. Yes, the face of the person is all the way in the right corner, okay, up, the open mouth and the nose. So when it is stuffing, that pathogen is coming out, okay, respiratory pathogen emission. So please go on clicking. There are a few things in the, hello, go on clicking 3 times. Right. So when a person is coughing, you can see next to his mouth is a lot of big droplets, large droplets, which cannot move more than 23 inches or roughly 2 foot. And the small droplets which can go like 4 feet or so. And further small droplets which can also travel even for 7 to 8 meters. So that's why we should have distancing. So that's why we say social distancing. We should be away from the positive person or affected person apart, and the mask and nose as well as mouth should be covered. And if we have typical proper mask, even that person cough, the virus will not come out in the vicinity or in the air, because it's more like aerosol and it's floating and stays for a while. So next, please. Yeah. But I will very simply, the first left one, the upper one, why with component and inside the RNA, so that virus, when it is entering, during that time, people are using neutralizing antibody, okay, to protect the host. And all the receptor, which is AC2 receptor. And the other receptor, which is TMPRSS2, which is transmembrane serine protease, people are also trying to get inhibitor for those. And nowadays, a lot of structural biologists, medicinal chemists, they are using different software, trying to find out what could be the best one. And finally, in the laboratory, we are trying to synthesize those molecules which can inhibit those receptors. So that's at the entry point, we can use the drug. And once it entered, the endocytosed, they are also enter into the endosome. And during that time, pH is low in endosome, and the lipid bilayer is dissolving, RNA is coming out. Now people are also trying to use chloroquine. So chloroquine or hydroxychloroquine, that basically used as inhibitor for those membrane fusion. It don't allow those things properly. So that's why people, once it was a big hype that America wants more of the hydroxychloroquine just to use rampantly, but which is also not good. Depending on the condition of the patient, you use chloroquine or not. That should not be kind of preventive. It's rather once disease is there, then only according to the condition, chloroquine should be used. Okay, now virus is entering into the endosome, and now the RNA comes out. RNA will make another RNA. While another RNA is producing, then the RNA-dependent RNA polymerase, if we can inhibit that enzyme, and yes, those nucleoside analog is using, and that is nothing but remdesivir. So this is now very much using in the treatment. So our host system always trying to combat. So immune system is also boosted up, and some interleukin, some other chemokines, those are also started releasing, and chemokine burst is also there. So now you can see when RNA preparation has already been there, and new virus particle is preparing inside the cytoplasm, ready to go exocytose. Over there also, people are trying to catch hold, not to make the assembly properly, and that's why they are also using some inhibitor. I'll show you what are all these different drugs, because in February, when it started, the disease, we do not know much about how to treat patient. But after 4 months, a lot of doctors, what are those medicines and what to give and how to. So that's why our rate of recovery is really getting much better, which is definitely a very positive sign. So please go to the next slide. Next, please. Next, please. Okay, so our body has, no, the previous one. Yeah. So we have innate immunity as well as adaptive immunity. So both these immune systems are up following viral infection. So following entry of virus inside the host, it is recognized by pattern recognition receptor or pattern recognition molecule, which we call PRR or PRM. So those molecules are nothing but toll-like receptor. So those toll-like receptors are present in the epithelial cell of lung. So those TLR7, TLR8, and few other molecules like RIG1, which is retinoic acid inducible gene 1, and another one NLR, which is nucleotide binding domain and leucine rich repeat acting receptors. So big name NLR, RGI1, TLR, those are all expressed in epithelial cell. So it's as a immune response. And those pattern recognition molecule that recruit the adapter protein once it binds to the virus, and then activation of some other transcription factor, and also NF-kappa beta FP1 that's also produced, and some other interferon and chemokines that's also produced. These are all basically immune response after viral infection. Another set of innate response is like polymorpho nuclear lymphocyte, monocyte, they also produce more of those proteins like IP10, which is interferon gamma induced protein chain, monocyte proteins. So those monocyte chemotactic proteins, so those are also upregulated. And then a lot of lymphocyte come. So this is all immune response going on. The next slide, please. In the next slide, what we are doing immune response, where the between innate and adaptive response is critical for clinical progression. Means T cell is produced, protective response is T cell dependent, with CD cells geared towards production of neutralizing antibodies and toxic CD8 of eliminating infected cells. And there are some others, CD26, which is amino peptidase, that's also involved in T cell activation, may bind to the spike protein of COVID-19 and resulting in a non-productive T cell infection, which is also still we don't know about that induced cell death, AICD. The other CD147 is also immunoglobulin superfamily, which induce metalloprotease, matrix metalloprotease, which is, can bind to S1 domain and facilitate viral entry. And in severe cases of COVID-19, T cells are functionally exhausted. So these are all the different immune systems that's start going on after viral infection. Next slide, please. So there is a delayed type 1 interferon response, and viral replication triggers a hyperinflammatory condition, an influx of more of the neutrophils, inflammatory monocytes, and the T helper, Th17, those are also induced because they produce the specific antibodies. So you can see in the figure, in the lung epithelial cells, those at the initial time, the type 1 IFN is there, and then more of the plasma cells, this B cell produce antibody, T cells also produce more of the cytokines, and macrophage neutrophils, they get activated. So this is all the responses, immune responses in the host. Next one. So this picture shows how the other two viruses like SARS-CoV-1 and MERS-CoV, how the treatment was. The treatment here you can see panel when SARS-CoV-1, so people use the drugs which basically inhibit the path for inhibiting those TLR mediated immune responses. So they use inhibitor for PKR, DPK1 and IRF, and for MERS1 also they used the similar mechanism. With the same mechanism can be used for new CoV, SARS-CoV-2. So that's the experiment going on with little bit of modification of the earlier drug people have used. Next one, please. So here you can see the inflammatory response through monocyte and macrophage. When that uninfected monocyte or macrophage from bloodstream invade the lungs, where they detect virus particle and a nuclear component, in this immune complex, these particles are taken up into the cell. Here you can see the receptor that I already talked about, ACE2, and then they are presented to the TLR and activating the transcription factor NF-kappa beta, and as a result, uninfected monocyte and macrophage produce significant amount of cytokines. So we can think of inhibiting the IRF or IL-6 for combating COVID-19. Next, please. So these days, antibody against this protein is the major way to block the virus attachment, because people, those who are very susceptible, means positive these cells as angiotensin converting enzyme positive cells. So there the virus attachment is going on, and antibody against S protein can block that. So once antibody is given to the system, we know IgM is produced followed by IgA, and they detected early during first week of symptom, whereas IgG can be detected after 2 weeks after initiation of the symptom. So still today we do not have much knowledge how long the protective effect remains. Means now we are thinking we should need vaccine, but we still don't know that vaccine that attenuated virus. So that vaccine will stay for 3 months, for 6 months, or maybe after 1 year you need different modification of the virus, just like other flu, which works in this year, the next year doesn't work. So those are still black hole. Next one, please. Yeah. So this is in that picture where, as I told, 5 days exposure, then the symptom comes, and if we give antibody, then RNA will go down. That means virus number goes down by 3 weeks. And in the meantime, in our system, we have IgM and then more of IgG, and that remains. That remains for 1 or 2 years still. We don't know, because that memory is how long it will be. Okay. But now there are different companies, they are trying their best, they are thinking it would really work. Still, whom to give, the children of which age, or all the people, or who are vulnerable, how, whether they have that much of strong immune power to take the vaccine or not, because comorbidity is another major problem in COVID-19. Okay, so next one, please. Immunological profile of COVID-19 patient. So in severe COVID-19 infection, we see that exacerbated pulmonary and systemic inflammation. I mean, more of the inflammation. And the level of CRP, which is C-reactive protein, gets up. Lactate dehydrogenase also get up. IL-6, that means cytokine storm is there. So that's one of the immunological feature of COVID-19 patient. And a few other interleukin, TNF alpha, T-naive cell, that's also get elevated. And those lymphocytes like CD4, T memory, B cells, are found down regulated. So please go to the next one. Regarding chloroquine, I have one of the cartoon slide. What you can see that the green receptor, the light green receptor, which in presence of zinc, it can enter, zinc ion can enter into that receptor. But if cannot enter, but if chloroquine is there, then it can enter well. And once it enters, then the RNA comes out. The cells membrane and binding molecule limit the ability of zinc to penetrate into the cell cytoplasm via special port. Okay, and this particular drug, chloroquine, activates the port and allow zinc to enter the cell. And now in the cell, zinc is then able to block the enzyme, which is RNA-dependent RNA polymerase, which turns off the viral replication. So that's why chloroquine is a kind of hype people are going to use, and they will be fine. Okay, next, please. And now biologists are also trying to make the simulation. The earlier slide where I'm showing that MFDC, how it is interacting in computer simulation process. Okay, this slide, what it says is potential therapeutic targets. So what are those drugs? What are those potential biomolecular target? Okay, as you know, nowadays people are thinking for plasma therapy, means convalescent patient, means those who already have COVID infection and they are recovered. So within 3 months, the plasma which already have the antibody, means memory, so that plasma can be given to positive patient and they will be fine. Okay, means virus particle may be caught and inactivated using antibodies from convalescent patient. The other potential therapeutics is soluble enzyme, angiotensin converting enzyme protein, because that can bind to the virus. The hydroxychloroquine, as I told, that can be used because the propagation can be stopped in combination with azithromycin. The antiviral treatment with protease inhibitor, there are lopinavir, ritonavir, those are protease inhibitor, because when the protein synthesis is going on using host machinery, there are few proteins that needs to be cleaved to become functional. Okay, they are non-structural proteins. So those protease, if we inhibit those protease, then viral assembly component, those cannot be done. So that's why those protease inhibitor is also a potential target. And RNA dependent RNA polymerase, what I already told, that nucleoside remdesivir, that is also used, can limit the replication. The other, when immune response is already there and interleukin 6, interleukin 1 is produced, people are now use the blocking signaling through receptor antibody, which is tocilizumab, and that limit the further immune activation and cytokine storm. So those are all these potential therapeutic target, and here in this red inhibitor sign, you can see all those different drugs that are using these days. Next one, please. Next one. Yeah, same thing what I told right now, at which point we can use all these inhibitors is basically same it's described here. Okay, next one, please. No, the previous one. The previous one. Yeah. So that important information for vaccine development and evolution should be well defined, because first we need to know the target antigen. We should know the immunization route, how we are going to give, orally or intravascular. Polio, you know, it takes long time for a vaccine to really work. Initially, it takes to make 2-3 years, and whether the things are working or not, to know those things 10 years, and to eradicate everything another 50 years. So it's not this easy job. So that's why immunization route and correlated immune protection, also what animal model, the scalability and the production facility and target product profile, outbreak forecasting and target population, collaboration and technology. So those are a lot of different parameters that needs to be looked into very critically before a vaccine comes and to apply to the mass. Okay, next one, please. Vaccine discovery, why it is so challenging? So challenging because it's RNA virus, and the is significant virus. So RNA is in it is 6 mutations per site per cycle, and it put 1 ml of sputum is 10 to the 7 virus. That viral genome mutates more than once per site. Due to such mutation rate, the genetic configuration of COVID-19 will vary region basis. What it works in America may not work in Israel. What works in here in Calcutta, that may not be good in Tibet. So those are all these regional variation is there. Next one. Very recent paper in PNAS, where they also looked into phylogenetic network analysis of this COVID-19 genome, SARS-CoV-2, and you can see there are 3 major strains that they found: type 1, type 2 type B, type C, and there are a little bit of mutation in B and C. So that's why this is challenging, because another new mutation will be there or not in future. That's also we don't know. So the antibody which is now produced against maybe these three may not work for the future one, which is going to be changed, and the old vaccine will not work then. Next one, please. So now I think I'm going to conclude my talk to say a few things, which is the major strategy to combat is vaccine. It is so many companies they are trying. There are different phase. Some in China, a few companies they are almost in the last phase, and I know in Oxford they are also going to sell their stuff maybe very beginning or in the next year. And the plasma therapy which is going on, but now ICMR is not really giving the permission for that. And the antibodies that we can make for these cytokines, that can be anticoagulant. I say a few words of anticoagulant, but uh, whether it's really a great drug, you can judge, because in Africa, South Africa, one lady doctor, when she's getting more of the patient, she found to put them in the ventilator, but she was curious to see the lung X-ray and found a lot of dots where the coagulation is there, and immediately she put aspirin or heparin, which can dissolve the clot, because for people those who are having heart problem, they also take aspirin. So using that anticoagulant, it works so well for those patients. They don't need to go to ventilation. And accordingly, she treated all those patients and they went home very nicely. So anticoagulant works so well for this. So nowadays, people are now using this anticoagulant for treating the patient. And herd immunity is really very futuristic thing, because more than 60% population, if they get immunity because of exposure, little exposure with all these COVID, they also start producing immunity against it, and ultimately the severity of the COVID-19 will be very mild, and people will be fine with very little drug like paracetamol and maybe aspirin. So herd immunity for a big country like India is very tough, because 130 crore people and half means 70 crore. And the Netherlands and Sweden, they are taking this one as the measure and to fight the COVID, and they are some success they've got. But by this time, we do not have much to do, only some supportive way, preventive measure. And that's, I will also say same thing. We should have social distancing, and which really through lockdown, that can very nicely be possible. My previous speaker already told that. Even the Swiss, they found less amount when lockdown is there. So social distancing is the way to really win the situation, and wearing masks definitely, so that those who are affected people and having some droplet in the ear should not come to you if you have mask. And you never know where you went, where you touched, and better in a regular interval you are washing your hands and face with soap. Soap means that detergent, so that the lipid bilayer will be dissolved, and RNA, if it is open, and RNA, there are plenty of RNAs in the hand, and it will be just clean. So that's why that's very good. And bleaching your rooms and sitting places in hospital and houses, that's also helpful for stopping the entry of the virus inside the system. I think I'm going to conclude. Next slide, please. Yeah. Next one. Next one.
So, thank you, Dr. Swarnakar, for your very enlightening talk. There are a large number of compliments for your talk from the participants, but there are also some questions, ma'am. So, can I forward the questions to you? Hello, ma'am. I think Dr. Swarnakar is probably not online. Could the host check, please?
Hello, madam. Hello.
Yeah, can you listen?
Yeah.
Please.
Madam, yeah, you have some, the participant have probably have some questions.
Sure, sure,
Dr. Yasmin will be forwarding it to you for answer.
sure.
Okay.
So, ma'am, Sanghamitra Purkayastha asks, how
Yeah.
is the efficacy of virulence different between symptomatic and asymptomatic carriers?
Asymptomatic carriers, they are not carrying virulent bacteria. Okay. Whatever they are carrying, that is less virulent.
Okay.
Mainly, but you know, in science there are exceptions. But
Yeah.
I'm not talking about exception, but as a whole, usually they are not very contagious.
Okay. Thank you, ma'am. Ma'am, another question is from Yahya Bakhtiar Laskar. So he
Sure.
asks, we have faced 3 coronaviral outbreaks in the last 15 years, and
Right.
considering the genomic variability or mutation rate in RNA viruses, especially
Yeah.
their rapid evolution, do you think a vaccine or drug could be effective in the long-term treatment of the infection?
See, vaccine would be a challenging because the mutation rate, if it is high,
Yes.
then now whatever vaccine is coming in market will not be 100% fruitful for the mutated one. Okay, so considering whatever vaccine, it will help 60%.
Okay.
Okay,
Yes.
then 40% is to take the drug according to situation or supporting medicine required. Okay.
Okay, thank you, ma'am. And ma'am, one last question. This is from
Sure.
Sukanya Purkayastha. So she
Sure.
asks, what do you think can go off target while dealing with different inhibitors? She's
Well,
asking about the off target effects of different
that's right. I understand. See, it's always a balance. It's always a balance whether immune boosting, the chemokine, all those chemokine producing too much. In that case, the drug is required. And now, even if it is have little bit of off target, still system can manage. Okay, so then the life and death problem comes. Definitely we will use like interleukin 6 receptor or interleukin pathway to block. Okay,
Yes.
because we won't allow too much of the immune boost.
Okay.
Yeah.
Okay. Yes, thank you so much, ma'am. So we are very grateful to have had you today, and we are very grateful to you for sparing some of your precious time for us. Thank you very much, and we
You are
hope
highly
that you will
welcome.
visit.
Highly welcome. I also enjoyed a lot, but little bit of problem initially for the slide, but the university helped me a lot. They changed those slides properly when I asked, and it makes my life easy. I enjoyed talking to and came to know that they listen to me and they have questions, and I also can ask a few things. Great,
Yes.
great.
Thank you so
Thank
much,
you.
ma'am. And we hope you will visit us in the future when things are
Sure,
better.
sure.
Thank you, madam. Madam Swarnakar.
I hope for
Madam.
herd immunity will come by beginning of October. Will be fine.
Thank you,
Yes,
madam. Take care.
Sure. Same to
Okay.
each of you.
Yeah. It is very nice to have you here. Okay. Thank you.
Okay, bye.
Bye-bye.
so with permission from the host, should we start the next talk?
Yeah. You start. Next one. Please invite next speaker.
Yes. Okay, thank you. So we know that in all the research that we do in our labs is finally translated to products in industry, and that is why we thought it relevant to have a speaker from industry who would address us today. I am very proud to present next speaker, Dr. Chaitali Bhattacharjee. So, having started her career in academia with a PhD in molecular biology, Dr. Chaitali Bhattacharjee continued her career in research with postdoctoral research at the CSIR Institute of Genomics and Integrative Biology, New Delhi, where she studied the role of microRNA in neuronal disorders. Subsequently, she worked as a DST fast track young scientist at the Sanjay Gandhi Postgraduate Institute of Medical Sciences, Lucknow. She was senior executive of Pancha Biotech Limited from 2010 to 2012, assistant manager WNS GSK Knowledge Center from 2012 to 2016, manager EvalueServe 2016 to 2017, and currently she is the lead client account management healthcare of Deloitte, US, India. I'm also happy to say that Chaitali Bhattacharjee was my senior during my PhD days, and she is a very close friend. So I'm proud to invite her to deliver her talk. Dr. Chaitali Bhattacharjee, please.
Hi, Yasmin, and hello to this
Yes.
entire
Hello.
crowd.
Hello.
Are you? Yeah, are
Can
you
you
able to?
please share your slides, please, with us?
Sure. 1 minute.
Yeah.
Can you hear me? Thing?
Yes. You
Okay.
are clearly audible. Yes.
Okay, that's great. All right. So let me just start. Let me know when you can see my slides.
Yes.
You can?
No, not yet.
Okay. I can see it is trying to share the content. Okay, there's some problem in sharing the content.
Okay, since you have shared your slides with us, so I think the host will be able to upload your slides. Sir, could you kindly, yes, upload Dr. Chaitali's slides, please?
Okay, so meanwhile, while the slides are loading, so thank you so much, everyone, for joining this session. Thank you, Yasmin, for this wonderful opportunity of getting back to an academic oriented audience, and it's really great to see all of you here. And the objective of this particular presentation that I'm going to share with you shortly, is the objective is really to give you an inkling about what the industry does, because on the whole, we know that, just like Dr. Yasmin had just said, that all the research that we do and all the research that you do ultimately translates into the industry, and that is where we can take care of the various diseases that afflict us. So the idea of the industry coming into this picture is very crucial in everything that we do, because we have to understand that the industry is a place which essentially helps us in taking this entire journey forward in finding cures for whatever afflictions that we are looking at. So for the purpose of this presentation, I would like you to imagine that we are in a baton relay race. The race where the baton is essentially passed on from one person to the another, and then finally we reach the finish line. And the race to find a solution for COVID is also very similar to a baton relay race. And if you talk about the players in picture, so we have the life science and the pharma sector, which does play a role there. Then we do have, the
Anyone else, any other speaker, any other participant, I'm sorry, who would wish to put forth their views? Raima Das? Anyone? Raima? Raima, are you online with us?
I cannot see her online here. She might be on YouTube.
Okay. Anyone else? Nobody is
Anyway,
volunteering.
that's all right. That's all right. I can understand. I mean, it's a sudden, and this was a sudden request. So I can completely understand. Uh, so okay, maybe what I'll do in that case is sort of give you an idea of what I do, because I was not intending to really speak about that part, and keep the topic focused towards the COVID situation. But yes, so I am currently with Deloitte. So Deloitte is so we have these consulting firms. So it is one of the big fours. In fact, it is the biggest big four. That is, it is the biggest, it has the highest revenue in amongst the consultant firms, and it is. So we obviously cater to each industry, every industry that you can think of. We cater to everything from say industry. I mean, you have life science, healthcare, you have financial services, you have technology, telecommunications, you name it, obviously. And markets which we serve, and this essentially is divided into two parts: life science and healthcare. When we talk about the life science segments, the life science segment is essentially the part which deals with these biologics that we talk about, the different kinds of molecules that you come with. So that is essentially the pharma and the life sciences sector. When I talk about the healthcare sector essentially takes care of the hospitals and the plans, providers. So these I know are words which would be alien. I wouldn't be surprised if these are alien words to you, because that is something that only once you are words which are typical to the industry. These firms that I support and make sure essentially help the entire working towards making. So there's a lot of studies that go around, and there's a lot of analysis which really goes around in determining what are the kind of services which will best cater to the client, and what is it that will cater best to the world at large. It's essentially what brings the money for the company and brings in the solutions for the people. So they, I've learned, go hand in hand. So the more money you make,
Bhattacharjee,
you essentially. Yes.
Dr. Bhattacharjee, if I may interrupt you, so your slides are now live. We can see the title slide. Are you able
Is
to
it?
view the
I
slides?
can't. I can't see the slides though.
Okay, so we can see the first slide. So as you go, you can, the first slide is your title slide. And as you want us to change the slide, you can just say next slide and we can change it for you.
All right. So, Dr.
Thank you so much.
would I be?
Yeah.
You're welcome. Would I be able to see the slides which are being presented? Because I'm not able to at this time.
We are not able. I don't know what is the option with the sharing, because we are able to.
That's okay. That's okay.
Yeah. Yeah.
All right. So
If
I
you
will
could,
just go forward.
yeah, the current slide now is your title slide, Dr. Bhattacharjee.
All right.
And whenever we have to
finished my
change the
introduction,
slides, you can just say next slide, and we will change the slides.
Sure, I'll do that.
Okay.
All right.
Thank you.
So, much, Dr. So this will go differently than what I had envisaged, but we will still, make this a very interesting discussion. So what I had intended to do was, first of all, get a feel of what you feel about the industries and how they are taking us forward in terms of the finding a solution for COVID. So let us begin with a poll. Can you go to the next slide, please? So I believe we are at the next slide. So I would just ask you, the participants, to please indulge me a bit and log on to this particular site called menti.com. Okay, so essentially you just have to open a new tab and just type menti.com. Okay, and use the code which is given on this slide on your, which is flashing on your screens right now, and just use that. Okay. Once you do that, you would be able to see a question. And what I essentially want you to do is just give me a feel of, just write your answers there. I was planning to share the results with you. Let me see if I can figure something out quickly as to how I can share the results with you. But nonetheless, let us just see what you have to, say. So while you are logging into this particular site and using the code, so the question will come up. And the reason why I am asking you this question is that because I am from academia and I know very, very well that, while we are in academia, we are slightly removed from the industry, and we don't necessarily have as much of knowledge or inkling about what is happening in the industry. That is why this particular poll, to really understand, and this will be for my knowledge and my understanding, to see what is the academia speaking about the industry, and what, according to the academia, is the company which is really going forward. Okay, so let me just see if I can. Yeah. Okay. I can see some names popping up. So yes, these are interesting, interesting answers. Yes. So yeah, yeah. Let me see. Yeah, so I'll just tell you the names which are coming up, which is, yeah, I can see quite a few people have talked about Bharat Biotech. Okay, people have spoken about Serum Institute. Someone has spoken about SmithKline Beecham, Lab Research, Pharma, Ghana. Bharat Biotech seems pretty popular here. And yes, it's gaining its popularity as I'm seeing the word growing. So let me just, yeah, I'll just give it a while. Okay. That's interesting. I will try my luck at sharing my screen once again. I think it will start. crossing my fingers because I want to see, how interesting this looks. But no, I don't have any luck in sharing, what I thought I would be able to share with you. So what I will do instead is, I will maybe, just tell you, what we have seen. So yes, Bharat Biotech has the largest number of takers. There are lots of people who are saying it's Bharat Biotech which will be winning this race. That you have AstraZeneca. Then some people have just said no idea. Yeah. So fair enough. So which essentially means that we do have some inkling about what's going on in the industry, which is great. But yeah, even then what we would like. So I would like to take you through the industries exactly, the companies where they are at their in their race, so that we understand where what the industry is exactly going to. Can I move to the next slide, please? So first of all, I will be talking about the vaccines and the repurposed drugs that the industry is using to fight this particular disease. So we have speaker after speaker today speaking about vaccines and how they have been, pretty successful.
Dr. Bhattacharjee, you are not audible to us. Hello, Dr. Bhattacharjee? Dr. Bhattacharjee, are you with us, ma'am? Hello, Dr. Bhattacharjee. We are not able to hear you. Dr. Bhattacharjee, are you online?
I am online. For some reason, can you hear me now?
Yes, we can hear you now. So would you like
Sorry,
us
I
to go
didn't
back
hear.
to the slides?
Yes, can you
The
please do
slides,
that?
yes. Yeah.
Which slide were you on? I have been just
Vaccines and
speaking.
repurposed drugs. Vaccines and repurposed drugs.
Okay, can you just move to the next slide then?
Yeah, sure.
So am I audible?
Yes, you're audible. We will just upload your slides again.
Yeah.
Yeah. So, Dr. Bhattacharjee, we are on your fourth slide now, drug discovery and development along risky road. Yeah.
Right,
Yes.
great. Okay, so apologies to all the participants who have been listening to me. I have been speaking on this, not realizing that the connect somehow the on off. But anyway, I'll just take you through this slide. So when we talk about the drug discovery and development, it is essentially this slide that we are. So if you see, we are talking about drug discovery, which is marked in blue, right? So this is the part. Then it moves to the clinical trials, okay, which is the part marked in green, and it continues till the FDA review and the large scale manufacturing. All right. So now let us look at the drug discovery phase. So we have 5,000 to 10,000 compounds in the drug discovery phase, of which, which is essentially the part of which I was a part of a few years back, and you guys are a part of now. From which we move to the preclinical phase, which is essentially the phase which I don't have to explain this to you, but when these particular drugs are tested in animals, From here, an IND is submitted and the drugs move to the clinical trials phase. The phases are phase 1, phase 2, and phase 3. When it moves through these 3 phases, that is when the drug is ready for the FDA review. And once this review is done, that is when we will consider that the drug we have an approved drug. Right now, there's a phase 4 clinical trial as well, which I will be covering in a while. Now, I would like you to pay a little bit of attention to the numbers. So you see that we have about 5,000 to 10,000 compounds which are in the drug discovery phase, from which about 250 molecules move into the preclinical phase, right? So amongst these 250 molecules, we have about 5 molecules which move into clinical trials phase. And from amongst these 5 molecules, you would have approximately 1 molecule that will essentially become an FDA approved drug, right? So that is the story in terms of the numbers, right? Now let us look at at this particular story in terms of the time taken. So the drug discovery phase takes approximately 3 to 6 years, right? Then we have clinical trials, which takes 6 to 7 years, and the review that takes about 0.5, I mean, that is half a year, 6 months to 2 years, right? Once this drug is approved and it hits the market, then what happens is there are certain cases where there is some kind of an adverse reaction to a drug, or the drug is not efficacious enough in terms of what we think it would be. That is when a phase 4 trial or a real world evidence kind of a study is launched to really understand if the drug is efficacious enough, and say there is some kind of a problem that happens, you're back to the drawing board. That's how the entire drug discovery and development phase essentially goes through. So you look at the number of years that it takes. It takes anywhere between 13 to 18 years to get a drug into the market, right? And this is obviously in the pre-COVID scenario. That is when we don't have some life-threatening situation. That is the time when we are able to make sure that we are, that there is that this is how the drug comes into the picture. With the COVID scenario, now I would like you to again take you back to the analogy of the bait and relay race, because you have to we must appreciate the fact that we are now asking the drug to be created in a year or less, okay? And so you can very well imagine that even a year or year and a half is stretching every possible limit, right? So let us now move to the next slide. Okay, for the in interest of time, I'm not really telling you what the phase 1, phase 2, and phase 3 clinical trials are all about. But suffice to say that in phase 1, we are only looking at the safety, if the drugs are safe enough. In phase 2, we take a small population, a slightly larger population, to see if it's efficacious enough. That is, if it has the effect that we desire it to be. In phase 3, the trial is done on a large group of people to see if it is efficacious enough or not. Okay, that's what it is. Can we move to the next slide, please?
Yes, you are on the next slide now, Dr.
Okay, that's
Bhattacharjee.
great. All right. So, okay, now when we talk about the COVID scenario, okay, now you can see, the syringe on the right represents the drugs that are currently under, under stage, so under trials, okay. And now you can see that you have this many drugs in phase 1, phase 2, phase 3, and the regulatory. You would obviously see that there are none which have reached the regulatory review, except for remdesivir, which has sort of reached the regulatory review part. Okay. Now, so there are, if you see, if in terms of numbers, there are currently 199 vaccines in the development for COVID. Okay, they fall into 9 different product categories or platforms, which range from the inactivated virus to live attenuated virus to DNA viruses to RNA viruses, you name it. So all kinds of vaccines are being tested here. At this time, there are 19 vaccines that are in one of the 4 phases of clinical testing. By the way, this is the data that I took day before yesterday, and today morning when I checked, the number has changed. So there are 20 vaccines that are in one of the 4 phases of the clinical testing. All right. So that is how fast things are moving. Also, now I would like you to like to take your attention to the small box. I hope it is visible to all of you. It's the writing is not very clear, but this gray line that you see is essentially the entire life cycle of this is the amount of time that it normally takes for a vaccine to come into the market. The blue line that you see below is the amount of time that the COVID will take to really hit the market, right? So this is how fast things are moving. I will take a step back here to talk about something very interesting which had happened in December 2019, which is WHO. Every year, it generally sends out a notification to all the and it sends its notification to all the companies and all people who are conducting research as to what it should really be focusing on. Okay. 2018, interestingly, WHO did something which it had never done before, which is it said that we must be preparing for a disease X. Okay, so it was just simply called a disease X. And if you categorize disease X, it would be something which would be viral in nature. It would have an RNA as its genomic as its genome. Then it would have a virulence of this much, everything. And if you match that with coronavirus or COVID-19, it actually has an almost perfect match, which also, if you have the time, I would urge you to watch a video which Bill Gates has actually uploaded some time back. And he had also predicted that something like a coronavirus, and it will essentially be a coronaviral vaccine, a coronaviral disease that will afflict the world. So why I am saying this is that so the inkling is that the industry was sort of preparing for something like this. So that essentially may have, because if a direction comes from the WHO, you seriously, and there would be some kind of preparation that they would already have been doing, and that is where maybe that is the reason why things are moving as fast as we see them moving. Right. So can we move to the next slide, please?
Yes, Dr. Bhattacharya, you are on the next slide now.
Yeah. Okay. Thank you. All right. So these are I had asked you these questions, right? And some of you have talked about these vaccines which are coming into the picture, and I can completely get. And by no means am I trying to say that this is what it will look like. And when I'm talking of a race, by no means do I mean to say that AstraZeneca, which is right on top there, is actually going to be the first to get there with the vaccine. Don't know. It's an ongoing race, and we don't know which is the vaccine which will hit the market the first. But yes, at this current moment, AstraZeneca does seem to be the company which has come up with its set of with its vaccine, which is essentially an RNA-based vaccine, which it is which has reached the late stages of phase 3 clinical trials, and this will hopefully hit the market sooner than later. Then you have some companies from China which are also very much into the picture. Also, this particular table is obviously not and it does not present the entire picture or of all the studies which are currently being doing. But then you can see Sinovac from China, which has come up with a vaccine, CoronaVac, is essentially about to which has just hit the phase 3 trials. Moderna has also, the vaccine, which is it's right there for you to see. It is an mRNA vaccine, which is about to, which has just entered the phase 3 clinical trials. And you can see that one after the other, where they have essentially, they are going forward with it. Bharat Biotech, many of you had written about it. Yes, it's very true. So they have created their own vaccine. It is somewhere in the phase 1 clinical trials right now. It means that we still have some way to go before we have an efficacious virus. Biocon, another company, Indian company, which features in this list, has come up with its, drug. It's not really a vaccine, but a drug. It's an antibody, which is itolizumab, which is about to, it has completed the phase 2 clinical trial. However, there are lots of concerns which are being raised about the way in which this particular study has been done, because the population size that has been taken is pretty very small. It's only and it is not considered significant enough. So they are fighting it out, and we'll see what happens. So this is, in a way, the way that you can see that the entire race is going on, and sooner or later one of them will touch the NDA approval part, and then we will have the drug in picture. All right. Now, can you please move to the next slide? Okay.
Yes, we are on the next slide, Dr. Bhattacharjee.
Great,
Yeah.
thank you. So now we are talking about some partnerships have also emerged out of this particular, this race to find a vaccine for or a cure for this particular virus. So we are talking about Operation Warp Speed here. It has a very fancy name because obviously it comes from the US and they only have fancy names. So it essentially means, what they essentially do is they have okay, so when I'm talking about the clinical trials and how long it takes, for things to really come into the picture, to come into the market, part of the reason, and a large part of the reason, is because there are a lot of a lot of scrutiny that goes in. Okay, for instance, if you are designing a clinical trial, you would have to get the entire study approved by the clinical trial board, and that takes time because you will have to wait for the other drugs who have put in their papers before you, and you have to wait for them to get their stuff done, and that's how it takes very long. However, Warp Speed is trying to make sure that we are able to circumvent some of the steps which could be done simultaneously, because if you really see, there are a lot of there are a lot of drugs there which essentially don't need to through such in such a situation. You cannot have a lot of debate about the study design. For instance, you cannot have a lot of debate about what kind of people are you enrolling for a study. So those things that they are trying to take care of. Also, they are trying to see if how many things are they able to do together. Okay, so maybe if a regulatory approval is required and 2-3 steps can be done side by side, they can do that. So they have essentially what they have done is, so this is Department of Defense, then you have the Department of Health Services, of which you have these particular bodies. Then they have all come together, and now they are making sure that they are taking care of this entire process. Yeah. All right. So
should I go to next slide? Would you like us to go to the next slide, Dr. Bhattacharjee?
No, not yet,
Not
Dr.
yet?
Chaudhary.
Okay.
Just a second.
Sure.
Yeah. Can you give me a second, please? Yeah.
Yeah, sure.
All right. Dr. Chaudhary, can you hear me?
Yes, yes, Dr. Bhattacharya, we can hear you
All right.
clearly.
Okay, thank you for that. My battery was running low. I had to get the charger. Anyway, so then the partnership that I would like to talk about, which is the COVID Plasma Alliance, which is essentially is the very similar thing which is being done in India, which is they have essentially brought in companies which have come together to make sure that they collect plasma from convalescent patients and use that to see how we can get a drug out of this. We obviously know that this is very, very difficult to achieve. Against this particular virus is difficult because of the high mutagenicity of the virus, and that is why maybe a plasma therapy is not such a bad idea. But having said that, it is still it is a long shot, and considering how fast we are being asked to move, that it becomes extremely difficult for us to see what it is. Okay. All right, so can I just move to the next slide, please?
Yes, you're on the next slide now.
All right, great. Now let us look at the repurposed drugs that we are looking at. So here you will see, you have remdesivir, which has been, talked about endlessly about how, etc., etc. Honestly, it is if you look at the studies, it is the are doubtful, not really doubtful, but the efficacy is not as much as it is being made out to be. But yes, and to top it, it is prohibitively expensive. So if you are to take doses of remdesivir, it is so expensive that you cannot expect this to be a drug of mass use. So either ways, it does not make a lot of sense. The Bosch Health has come up with the Virazole, uh, which was being used for lower respiratory tract infections, and now this is what has happened, and they are trying to see, it has essentially reached phase 2 clinical trials. Then you have Novartis, which has its plethora of drugs. Three of them are listed here, which have all reached phase 3, and we're hoping to see something out of that. And these partnerships, like Regeneron and Sanofi, were working with a drug called Kevzara, which essentially affects is a drug used to treat severe active rheumatoid arthritis. And if you see that it reached the phase 3 trials, and then what happens is that you find that the phase 3 the trials are not efficacious enough. It is not showing the results that you would like it to be, and that is where the entire story stops. So they obviously had a lot of hopes there in terms of bringing this drug to the market, but you can see that this particular drug will not be coming into the market. Similarly, lopinavir, ritonavir, this is also a very recent development, where this has also failed in the phase 2 clinical trials, and we have to see how this story progresses. But as far as it looks like right now, is it will not be progressing very far. So it is in this way that the life science industry and the is really working towards finding a cure for COVID, by number one, finding a novel cure for vaccines, and number two is rummaging through the drug repositories, trying to see what do they have that can be used to fight this particular infection. All right, can we move to the next slide, please?
Yes, we are on the next slide. Yeah.
Great. So now now let us talk about testing, right? This is something that there are companies which have come up with tests, and in a remarkably short period of time. Yeah, to be fair, it is relatively easy to find a test than to find a cure. So yes, but it's not that testing doesn't come with its own challenges, because finding the right test which will give you a positive result 99% of the time is no mean task, and that is what these companies have been trying to do. Of course, we have so can we move to the next slide, please? So
Yes, yes.
yeah, great. So for COVID-19 testing, if you see, we have the PCR, which all of you know. This is highly simplified. I know that this must look like very juvenile to most of you, but yeah, it is what it is. I mean, so yes. So when we talk about the polymerase chain reaction, we're obviously talking about qRT-PCR or RT-PCRs at max. So which essentially means that so if you look at the pros, yes, they are very accurate. Cons is that it has a long processing time, and another cons is that it is expensive. So not everybody can get a PCR test done, right? Now then you have antigen testing. The nasal swab that is essentially taken is something which is very rapid, and it is inexpensive. I think it takes a few hours to get the test results done, or even less than that. Cons is that there are accuracy problems, because antigen testing has its own kind of so if a person is positive, that person is definitely positive. But if a person comes out as negative with the antigen testing, it does not necessarily mean that the person that is COVID negative, because it essentially means that, number one, the person is perhaps not does not have enough viral load that is coming up in the antigen testing. Number two, is not in the correct phase of the COVID testing, because essentially the antigen testing works well when the person is about 7 days into infection. So you can well imagine, if a person is 2 to 3 days into infection, or even 9 to 10 days into infection, it will not test it. Okay, so that is where the antigen testing is not considered that useful. But it definitely, one positive side of it is that if somebody comes out as positive, the person is positive. This essentially means that you are considering a population as large as any country has. If you have antigen testing, that essentially reduces the patient load that needs to be tested by 50%, which means that there are only the 50%. So if a person has come out negative, you can send them for the PCR test or an antibody test and see what results come out. That makes it worth its while, and that is why antigen testing is still going on so much. Then we talk about the antibody testing. Antibody testing is essentially a way to find out if a person has been exposed to COVID before or not. It is rapid, and it can detect the disease spread really fast. Cons is that you do have false positives and false negatives. Another, when we are doing these antibody tests, essentially it is really something to be studied later, to really understand, okay, how many people got infected and how many people recovered from it, kind of a thing. uh, it has not been conclusively proven yet, for instance, that a person who has been infected with COVID once will not get infected again, which obviously stems from the fact that COVID is so highly mutagenic that a different strain, when it affects the person, might fall sick again. So hence, this does not have any conclusive evidence or proof. But yes, we can say that this will be an important test in moving towards herd immunity, when we see that a large population does have these antibodies that can help us to some extent. Okay, but at this point of time, it is mainly on a study basis that we can use these results for. If a person that has antibody, doesn't mean that person can go out. Not yet. Yeah. Next slide, please.
Yes, Dr. Bhattacharjee. Yeah.
Yeah, great. All right, so let us look at some of the tests which have come up. So Abbott has come up with a test called ID Now test. It's an isothermal nucleic acid amplification. This is pretty fast, and it detects the presence of. To come up with another sort of a test called M2000, which is also an RT-PCR. It helps in detecting the presence of infection. Both these tests, however, you have to be very, very specific. So these are error prone. So if anything goes wrong, the test results might go haywire, and that is something which is not great to hear. But then at the moment, these are leading in terms of the testing that is going on. Thermo Fisher is another company which has come up with TACPath. You well imagine that this is based on RT-PCR, and it also detects the presence of infection. Then you have Boehringer Ingelheim, which detects the presence of antibodies. Okay, then you have Agilent. Agilent is creating platforms to automate the steps for assays. So essentially, it is trying to create Agilent. Some of you might have used Agilent platforms. Essentially, they are these platforms to make sure that you are able to do your assays, a large number of assays together. So they are trying to make sure that that particular study is that particular assay becomes better and better. Then you have other companies like Christus, then Sanofi. If you had spoken about Sanofi earlier too, so Sanofi, in addition to working on creating vaccines, is also creating a smartphone-based solution where you can do a home test. So yes, so these are the various kinds of tests that are doing. In addition, next slide, please.
Yes, Dr.
Yeah.
Bhattacharjee, you are on the next slide.
Yeah.
Yes.
Thank you. So yes, then you have companies like Roche, like Cardinal Health. Okay, so these are all companies which are coming up with different kinds of tests to make sure that we are able to identify the presence of either antibodies or immunity against COVID. We have finally started testing a lot of samples. So hopefully, we will be able to make sure that we are able to detect as many patients as possible of COVID and help in containing this particular disease. The next slide, please.
Yes, Dr. Bhattacharjee. Next one.
Yeah. Okay. So yes. So the next player in this entire patent relay race is the medtech industry, because the medtech industry it has an equally important role to play. So if you observe the movement, we are moving from being in the lab to creating medications to testing, where we are going towards real people and testing it out. Now we are moving into a little more into maybe a hospital setting or something, where we are trying to see how these players are helping. So we have the next slide, please. So we have various companies, like you have Becton Dickinson, Medtronic, Stryker, Regeneron, etc. So all of these companies are essentially medical technology companies. So all these equipments that you see in hospitals are created by not exclusively by these companies, but companies like these. So here you will see that they have created things like tests, very rapid tests. Then you have systems. Okay. And so Becton Dickinson is essentially working towards making sure that they create the equipment or the devices which will help us do those tests, right? So you can see the collaboration between them. So only when they will create the tests, and these guys will make the equipment that is required for the tests, and then once that is ready, so they will combine the two, so the tests will go into these particular devices, and that is where it will go out. A Striker is an interesting company which has come up with an emergency relief bed. These are very very cheap, okay, because they just saw that we would be needing a lot of emergency beds, and so they just found out a way to create these emergency relief beds. Ventilators, obviously, right? That is something that everybody has been talking about the lack of enough ventilators and so on and so forth. So Medtronic come to Regeneron, which is come up with something like a Velocity suite, which helps the tests to go really fast. Uh, Univercells plus Excite is an interesting combination, because they are coming with prefabricated vaccine facilities, which essentially means you would have the vaccine facilities would be created in such a way that the production goes on much faster than before. So this is something that they have just started. Agilent also. So Agilent, Illumina, these are all companies which essentially make these platforms. Okay, and Illumina obviously is helping in sequencing the COVID and identification of the target. So this is how the entire thing is coming, story is unfolding. Can we move to the next slide, please?
Yes, you are on the next slide now.
Great. So now we are talking about the healthcare systems in COVID. So when we are talking about the healthcare systems, we are obviously talking about the doctors and the payers, providers, etc., which are typical to a US scenario, not in India so much, but nonetheless. At this point of time, I would like to you to imagine the situation of the doctors here, because so far they have nothing in their hands. Okay, they don't have a cure. They don't know what to do. They cannot protect themselves well enough because they I mean till some months back, okay, maybe 1 or 2 months back, enough PPE kits was a problem, right? So these are the people who are at the forefront of everything, and they are trying to fight this disease at best as they can. And you can imagine them as the last player in the baton relay race who's waiting for the baton to be passed to them. Once the baton is passed to them, they can rush through the finish line and make this entire story disappear. Unfortunately, at this time they don't have that in their place, so they are doing the best that they can, that is provide enough care. Okay, so let us see some of the things that they are doing. Can you move to the next slide, please?
Yes, you are on the next slide
Right.
now.
Okay. All right. Thank you. So you have these companies. I'm pretty sure would be would be companies that you would not have heard of before. So these are essentially healthcare companies. So they are the ones who say hospital settings at this point. That let's suffice to say that. So you which is a very leading, healthcare provider there. So they what they have started doing is they at no cost. Okay, then they have self swab COVID testing. So that is something that they are providing. That okay, fine, you go, you test, and you tell us, if you have COVID or not. Okay, because the US health system, as we know, was stretched to its very limit, right? Then you have the testing. Then you have Kaiser Permanente, another very big player. They have started with a COVID-19 vaccine trial. If you recall, there was this the first lady who received a vaccine shot. That lady received her vaccine shot from a company called Moderna, and it was done in a Kaiser Permanente facility. This particular vaccine has now reached the phase 3 trial. I had talked about it earlier, but nonetheless, this is what they are doing, and they're donating highly to make sure that the spread of COVID-19 is prevented. That is what they are doing. Adventist Health. Then you have Providence and Joseph. So these are all companies, and in fact, if you, I mean, a month back, when you would go to their websites and see, what they were doing, it was really it's heartbreaking to see, because they are talking about that okay, we are trying to make sure that we don't need enough PPE kits. Okay, we are trying to make sure that our doctors are safe. We are trying to make sure that people are testing enough, or people don't fall sick enough, so that we are not able to so that we don't have to really take care of this entire thing. And this is in a situation where we think that we've made so much of progress with healthcare, with so much of progress about finding cures for diseases and so on and so forth, but apparently not. So this has been a reminder to the entire industry and the entire world at large that we are not prepared, plain and simple. But we have to be better prepared in order to make sure that we are able to do it. And we should be able to get to the end of this entire journey and make sure that we reach where we want to reach, and this entire COVID ordeal is taken care of, and it's over from our lives. This is, in very short, what I had to speak about regarding this entire COVID scenario and the life science and healthcare industry's role in making sure that this is taken care of and we reach the finish line. Next slide. Thank you very much.
Thank you very much, Dr. Bhattacharjee, for your very illuminating talk with a vast range of information. So there are a large number of comments and lots of appreciation for your talk. And there are also a few questions. Can I forward these questions to you?
Yeah, sure.
Okay, so the first question is from Paramita Bhattacharjee. She says that there are these days there are a number of advertisements for immune boosting drugs to tackle COVID-19. Do these immune boosting drugs also go through testing and clinical trials in the same way as other drugs in order to determine whether they are effective?
So essentially, every drug that you have will have to go through, these clinical trials. So immune boosting drugs essentially fall somewhere in the range of not pure drugs per se, but they are more towards so for instance, there are a lot of these ayurvedic products that you would see which are immune boosting, right? So if in these cases they don't necessarily have the entire clinical trial phase. Okay, so though clinical trial is required, but it is not a very big mandate for them to go through a clinical trial. But strictly speaking, they should be going through clinical trials.
Okay, thank you. The next question is from Momita Nath. So she says, recently a team from Israel reported an audio test for COVID-19. Can you explain how an audio test works?
Audio test? I'm sorry, I'm
Yes.
not aware of that. Yeah.
Okay. Yeah. So we move on to the next question. The next question is from Juri Devi. So her question is plasma therapy really beneficial for corona positive patients?
So I'll speak logically, because at this point of time, none of us know what is really beneficial. But considering, okay, if a person is infected with COVID, okay, the plasma will essentially have antibodies against COVID, right? So that is the entire idea behind this entire plasma therapy that we are talking about. So if you take the idea is, if you take a large enough population of convalescent plasma, right, they would have these different antibodies, okay, and these antibodies would help in fighting COVID. So that the results, however, are has obviously to go through a lot of scrutiny, because it does not essentially there are other risks involved when you put in plasma, and because we are in such a tearing hurry to find a cure for it, we are obviously cutting corners there, and that is where the entire thing is. But logically speaking, it makes sense to say that plasma therapy should work. That is why you have this entire COVID program that I just talked about, right?
Yes, thank you. And we will take one last question. So according to this participant, as per our previous speaker, herd immunity can be one of the ways to combat COVID-19 situation in small countries. What is the future of COVID-19 drug industry in such countries?
I'm assuming you want to say that if once a herd immunity is achieved, what happens to the drug companies, right?
Yes.
Okay, right. So that's a very interesting question. So yes, herd immunity is something that everybody has been talking about, and I have my personal it's just my opinion that when we talk about herd immunity, it necessarily does not mean that all of us will just become immune to COVID one fine day because all of us have been infected with it. That is still under study. We don't know. So that is why I just mentioned, right? So we are looking at the antibodies that the body is producing. Okay, and we are only getting an idea about whether the person has been exposed to COVID earlier or not. But there are
Yes.
multiple reports which says that people who have had COVID before, right, are getting COVID again. So essentially, we don't know what that means, right? We
Yes.
don't know if the so there could be multiple things there, right? Number one is that it's a different strain of COVID that has infected the person. Number two is that the COVID was only dormant in the body and we thought the person was cured and now has come back with a vengeance. It could mean that. It could mean
Yes.
so many number of different things that we don't know for sure. So herd immunity is something that people have toyed with, countries have toyed with, and not done so well on that. So yes, to put this question, and the right thing is, like, I would say that I would say the life science companies and the healthcare companies are taking a very calculated risk because they don't see the herd immunity coming very soon. So they will make their money. Don't worry about that.
Okay. Okay. Thank you very much, Dr. Bhattacharjee, for your talk, for spending your valuable time with us. I apologize to you for the technical glitches, the
No
difficulties
problem
during
at
your
all.
talk, but we are very grateful to you for giving us this opportunity to listen to you. Thank you very much.
Thank you so much. Thank you for this wonderful opportunity. Thank you.
And to the participants, I would like to
Thank
say
you.
that we come to the close of the technical session with this talk. Following this, we have a concluding session, which will be hosted by our organizing secretary, Dr. Ravi Rajvanshi. I request all of you to remain online for the concluding session. Dr. Rajvanshi, please, over to you.
Thank you, Dr. Yashmi. Am I audible to all?
Yes, sir.
Yes, you're audible.
You are.
Yes.
Okay, thank you very
Yes,
much.
yes, yes, you're audible.
So finally we are coming to the end of the webinar, the entitled Approaches to COVID-19 Prevention Therapeutic Strategies and Management, organized by Department of Biotechnology. We had 4 speakers from India as well as from abroad, and we had really good lectures, talks from all the speakers, which were really enlightening for all of us, all the participants. The first talk was given by Professor Sushant Banerjee, who is from University of Kansas, USA. He delivered a talk on early warning and pandemic tracking of COVID-19 using sewage and wastewater. He also gave details regarding the viral genome of the COVID-19 and other viruses. Apart from that, how epithelial production is affected by the COVID-19. The second talk was by Professor Ariel Kushimaro from Israel. He basically gave a very good picture of many social events organized in Israel as well as Spain. At the same time, he highlighted that the social distancing is compromised during all the festivals, so we need to take care of the same. Describe the COVID viral genome as well as the techniques for the detection of the same by different techniques. And he also shared few examples that how this detection has been conducted in Israel. The third talk was by Dr. Snehasita Swarnkar from ISCB Kolkata. She delivered a talk on important strategies to manage and combat COVID-19. She delivered really a detailed, very good presentation on the structure of coronavirus. She also discussed the regions which are behind the sickness caused by the virus to cause infection, lack of immunity, which is one of the major reasons for pandemic we are facing these days. She also discussed about the life cycle symptoms related to the COVID-19 infection. Apart from this, she highlighted the clinical progressions and transmissions, details of immune responses during COVID infection proposed, which are proposed immune response during COVID infection. the mode of action of certain drugs like chloroquine. Apart from this, she also emphasized on the parameters that need to be looked into before the launch of the vaccine. And the last talk was by Dr. Chaitanya Bhattacharya. She has given a very nice picture of what the present scenario of the drug discovery and the development process. She emphasized and showed how actually this process is time consuming, which takes around 13 to 18 years normally for a drug to reach a market. And she told the data, she discussed the data regarding what is the present scenario. Like 199 vaccines are under developmental phases, around 20 vaccines are in the fourth phase of clinical testings, and some of the drugs which have also reached to third phase of testing. And she also discussed all the pros and cons of the COVID-19 testing and diagnostic methods. So these were the certain highlights of the talks, but as a custom, I'm supposed to give the vote of thanks to all the organizers. To start with, we have started today's webinar with an address from our chief patron, the Vice Chancellor of Assam University, Silchar, Professor Dilip Chandranath, who has been a very much motivating force for all of us, who has helped us to and provided all the necessary support to organize this webinar. Our patron, Professor Supriya Chakravarti, he is also very energetic and helpful always, and we are happy to listen him today. That he is coming up with a publication very soon, and that will be a contribution from the Department of Biotechnology to the present research of COVID-19. Our HOD, who is the chairman also for the present webinar, Professor P.B. Majumdar, was the main person who has taken all the pain and all the efforts to organize this webinar with such a grand success. The thanks also goes to the conveners of the webinar, Dr. Mahua Singh Gupta and Dr. Yasmin Chaudhary, who have taken so much efforts and organized the webinars with such a great success. Even though there were certain technical glitches, but they managed to somehow handle the situation very nicely, and the show was successful. The speakers, we are thankful to all the speakers, Dr. Sushant Banerjee, Professor Ariel Khushamaru, Dr. Snesikta Swarnkar, and Dr. Chaitali Bhattacharya for sparing their valuable time and for enlightening us with the present scenario regarding research and whatever the diagnostic progresses has been done in the same field till date. We are also thankful to all the faculty members of our sister department under the School of Life Sciences, as well as the departments, other departments in Assam University, as well as all the participants who have joined us from India as well as abroad, whose motivation has actually made this webinar a grand success. Special thanks also goes to the Director of Science, Dr. Anshuman Sen, and his staff, who has helped us a lot to provide all the technical facilities during the webinar. And last but not least, the special thanks also goes to research scholars who have put a lot of energy and efforts, especially Mr. Uday, Mr. Donald, Mr. Yahya, for their all the help and during the webinar as well as during the preparation of the webinar. So with this, I would like to take permission from the chairperson so that we can end the webinar. Thank you.
Very special thank to organizing secretary Dr. Ravi Rajbanshi also for his effort. And it is the joint effort of the department. I acknowledge all the colleagues our research scholars, all the university family members, all the technical experts, and other service providers, if I forget to name some. So with this, we can conclude, Dr. Rajvanshi.
Thank you, sir. Thank you, sir.
Okay.
Okay. Thank you. Thank you. Thank you, everyone. Thank you, one and all.
Thank you, sir.
Thank you, Yasmin. Thank you, Yasmin. Thank you, Supriya.
Yeah, thank you. Thank you. Yeah.
Thank you, Mohua. So, see you next.
Okay, thank you.
Okay, bye-bye. Bye-bye. Bye-bye.